Sure. But yeah, you also, yeah, more together. Once you organize the right online. Yeah, yeah. Yeah.
May I have your name, please?
Pachholz, P-A-C-H.
Is
Is
it in the afternoon session?
Yeah, afternoon session, right.
Okay, I'll just pick it up. Here it is.
Yeah, Andreas Pachholz, here this one.
All right.
And then I have a new.
Oh, you want me to update your?
Yeah.
Just a second.
This you can close.
Yeah.
This is one, right.
Okay. Put it in the afternoon session. And I delete the older one. Yes. Okay, cancel. I'll close the. Yeah, there you go.
You may have a look whether it's.
Sure, yeah, please.
Whether it's working or not. Flip through it. Okay, so I think the writing is big with mine. Just wonder how good. Look what it looks like. Oh, okay, it's big. No, it's good. So people can really read it. Okay, very good. So it's good to read. Great.
I'll eject your pen drive.
Yeah, okay, thank you. Yeah, klar, gerne. Please wait for his presentation.
Sure. There you go. Do we have our first speaker in the auditorium? Deepak Shiva.
Do you have kind of pointer?
We do.
Doesn't really work with the water point.
Yeah, so
Okay. Laser pointer. Sorry?
You can't really see it in the virtual.
So what I suggest you do is There's a point of
But
view.
you can also walk. It's so.
My presentation. Okay.
This is, but this is tomorrow.
Oh, it's tomorrow.
Yes.
Then we'll make it another floor.
Tomorrow live section.
Okay. Is it this one?
Yes.
Okay. What session was it?
Livestock.
Okay.
Let's just put the date.
Yeah, I will.
We'll arrange it.
Fair. Would you like to check your presentation or should I?
Yes.
Okay. There you go.
Okay. Presentation mode. Yeah.
Just
Great. Thank you very much.
You're
Okay. Genau.
All right, we're ready to begin now.
So what is you are thinking of our baby? No, directly. Launch it. Okay, so do it once and then.
Oh, thank you. I'll be sitting here if you need me.
I'm not going to just introduce.
Sure, yeah. We're ready for the first speaker as soon as they come. Great. As long as it works, can we change the slides here?
Works.
Yeah, it's easy.
So who is the speaker?
So you're speaking.
Yeah. I just introduce. So it's okay for mine? Okay. So good morning, everybody. Thanks for attending this pollution session number 2. And we will listen to Dipakshi Babar on national scale atmospheric reactive nitrogen budget from agriculture in India. So please, thanks.
Thank you for the introduction, ma'am. So good morning, everyone. As ma'am already introduced me, I'm Dipakshi Babbar. I'm a PMRF scholar and I'm doing my PhD actually I'm a second year PhD student and I'm under the supervision of Professor Srinidhi Balasubramaniam. ESED Department, IIT Bombay. The title of my presentation is National Scale Atmospheric Reactive Nitrogen Budget from Agriculture in India. So the motivation behind the study is imbalance in natural nitrogen cycle. As we all know that non-reactive form of nitrogen basically gets converted into reactive form of nitrogen in soil, and on denitrification, it goes back into the non-reactive form back into the atmosphere. But due to these anthropogenic activities like high temperature combustion in industries and vehicles, fertilizer manufacturing, and crop biological nitrogen fixation has introduced the reactive nitrogen into the environment and causing a cascading impact. For example, the N2O, as a result of high combustion released into the atmosphere, it has climatic implications. And because it's a greenhouse gas, for another is NOx from combustion activities or from the volatilization from cropland released into the atmosphere and causes the particulate matter like ammonium nitrate. Similarly, ammonia from the cropland, it leads to the formation of particulate matter. And the parts of the introduced nitrogen goes into leaches down into the it basically leaches down in the form of nitrate and found its way to streams or groundwater or coastal seas, and on further reaction it basically gets converted into other reactive forms and released back into the atmosphere, and on denitrification it releases N2 into the atmosphere. But this non-reactive form N2 which is released into the atmosphere is comparatively lesser with what we have introduced or created into the environment, like after fertilizer manufacturing and high temperature activities. So this released N2 is comparatively lesser. That means the more nitrogen is accumulating or circulating in the ecosystem. Talking about the cropland, as we know that population is increasing and the food, feed and fuel demand is also rising, due to which we need more and more crop production we have to do, and for which we are using more fertilizers. And this increased use of fertilizer is causing the release of NR into the atmosphere, into the environment, be it atmosphere or hydrosphere, and causing the cascading impact. In addition to that, UNEP adopted a resolution urging global effort to reduce nitrogen waste by 2030 and through improved sustainable management, and to share information on national action plans. But we cannot manage something until we quantify it. Thereby, there is a need to quantify all these NR emissions across various sectors in different medias. So the grand engineering challenge is to establish the nexus of 3 Fs, that is food, feed and fuel, with simultaneous reduction in NR emission. So it's an environmental dilemma to reduce the NR emission as well as to take care of food, feed and fuel security for the rising population. So this is the national scale budget. These countries have already established their national budgets, but for India, you can see that it is still yet to be quantified. Although impacts are already understood in the form of Indian nitrogen assessment report, but yeah, the quantification of the NR across various sector and media is yet to be quantified and understood. Thereby, my objective is to develop a comprehensive input-output accounting of reactive nitrogen flow as Indian nitrogen budget. But today, I'll be particularly focusing on agriculture sector. So here, based on the literature survey, I have found that these are the various economic sectors to track NR cascade, and different studies have found different sectors important in their country-specific emissions. Like for some have found cropland as the major contributor, for others livestock, energy and fuels, industry, wastewater, human settlements. Also, other sectors are also important, but for the country-specific scenario, these are the reported major ones. This is my methodology which I have developed. On the left hand side, this is the pool or sector, we can say. So I'm talking about cropland sector. Subsector is the nitrogen which we have added in the form of synthetic. So these are two categories. There are two categories: external drivers and internal drivers. In external drivers, the synthetic fertilizer, irrigation water, livestock excretion, nitrogen deposition, human excretion, and organic waste are the nitrogen input into the cropland. And for internal drivers, these are biological nitrogen fixation and straw recycled on the cropland system itself. The outputs will be in the form of crop, that is grain and straw, and other than that, losses, which are the various reactive nitrogen forms, be it ammonia, NOx, N2O, N2, or inorganic nitrogen, dissolved inorganic nitrogen. The media it is impacting is terrestrial media. So grain and straw has impact on terrestrial media, and these NR losses will be impacting hydrosphere and atmosphere. The analysis will be based on what is accumulated in the terrestrial ecosystem. Then for atmospheric emissions, it is compared with the existing database and with other countries' nitrogen flows, and also pollutant specific and process and activity specific indicators and drivers of rising nitrogen flows in Indian scenario has also been identified. So this is the overall picture I have developed to track the intersectoral nitrogen flow in different media and different multi-pollutant tracking in agriculture sector. And these are the tasks which I have followed to develop this overall picture, that is development of the framework, mathematical formulation, tracking the data as well as flows, input the data, define the fates of the output to avoid double accounting, and account the pollutant specific, that is direct as well as indirect emissions as a result of cropland specific activity and process. Find the missing parameters in the budget and balance the budget and compare it with the existing database. So this is development of environmental extended input output accounting model. It's a proof of concept. I'm taking fertilizer as an example to explain. So this is the boundary of cropland system, and in which the sum of all the inputs is equal to sum of all the outputs plus accumulation. These are the various inputs I have accounted for, and the mentioned are the data sources, but I will be particularly focusing on nitrogen consumption in cropland sector. So as an input, nitrogen fertilizer is added as a form of nitrogen in the cropland, and fertilizer multiplied by with the respective nitrogen content, because there will be different fertilizers. So respective nitrogen content, and the outputs will be in the form of what is accumulated, how much is taken up by the crop, and the losses. Now these losses have two fates, be it in atmospheric system or hydrospheric system. In atmospheric system, the fertilizer multiplied by the respective nitrogen content and the weighted emission factor. Based on that, I have calculated the emissions. So for ammonia, volatilization, that is the direct loss. For NOx, denitrification. And for N2O, three processes are responsible. That is denitrification, which is the direct loss of N2O, which causes this direct loss, and then leaching and runoff, and volatilization of NOx and NHX, NOx and ammonia, which causes the indirect losses of N2O. So these are all in atmospheric system. Then in hydrosphere, leaching and runoff leads to the dissolved inorganic nitrogen and causes this NR release in the hydrosphere system. Talking about crop uptake, how much nitrogen is taken up by the plant, be it in grain or straw form, is the crop uptake. And accumulation is calculated based on how much is input minus output. So to balance the budget. So talking about the inputs, we will follow the whole cycle in some certain subsections. So all the talking about all the inputs from internal drivers and external drivers, we can see that fertilizer, deposition, and livestock manure in external drivers, these three are majorly contributing highest nitrogen into the cropland system. And in internal driver, biological nitrogen fixation is the major one. And we can say that highest contributing reactive nitrogen sources are fertilizer application, biological nitrogen fixation, deposition, and manure application, which accounts for 96% of the total NR contribution in the cropland system. Okay, so whatever has been introduced in these various forms will be in two forms, organic or inorganic. Now these two, inorganic and organic, based on the different processes like dissolution, hydrolysis, mineralization, it will get converted into ammonium, and this ammonium has three fates now. It will either it will either gets converted into nitrate due to the nitrification process, or it will get assimilated in the plant, and or maybe it will get converted into ammonia form. So this ammonium or nitrate, whatever has been assimilated in the plant, is the 55% of the total NR introduced into the cropland system, and accumulation is less than 1%. So I can say that nitrogen use efficiency of the crops, because this study I'm particularly talking about 1 year, that is 2020, but I have done this analysis for 2 decades, that is 2000 to 2020. So nitrogen use efficiency of the crops has been varying from 47 to 55% over the years 2000 to 2020, and overall trend is decreasing. And earlier it was like 72 in before this green revolution in 1960s, it was 72 to 75%, but now this is the range. Talking about the accumulation is also decreasing. In 2022 it is less than 1%, with the high usage of inorganic nitrogen in the form of fertilizer, and also from deposition the soil has lost its efficiency to retain the nitrogen. This could be a possible reason. So 55% of the introduced is already taken up by the plant, 1% is accumulating in the soil. Now rest 44% has a fate in the environment. So this will be released either in the hydrosphere or atmosphere. Looking into the subdivision, hydrosphere is basically getting 25% of the total introduced NR. So runoff is the major responsible process for the NR release. And the rest of the 19% has a fate in atmosphere. And in atmosphere, there are three processes that is causing the release of NR, that is volatilization, denitrification, and obviously crop residue burning. But here I'll be talking about these two, volatilization and denitrification. So volatilization is the major responsible for 99% of the ammonia from the cropland sector into the environment. And for that, fertilizer application, manure application, and deposition are the three major factors responsible for the volatilization of ammonia. And we can see that on comparison, manure fertilizer is basically emitting twice the ammonia as compared to the manure, and half is coming from the deposition when we compare it with the manure. On denitrification causes the release of N2O, NO, and N2. Now N2O is released because of the two processes, direct losses and indirect losses. From N2O, from direct losses, we can see fertilizer application, biological nitrogen fixation, and deposition are the three major processes responsible for this N2O release. And this N2 and NO is also released during the denitrification. Now, direct losses are responsible for 86% of N2O release from the cropland system, and 11% of N2O is coming from indirect losses. It may be because of leaching, runoff, and volatilization of ammonia. And 63% of atmospheric NOx is due to the denitrification. Rest is coming from the biomass burning. So we can say here that 44% of the introduced NR has a fate to 90% is going into the atmosphere, and for 25% has a fate in hydrosphere losses. Also, volatilization is the majorly contributing process to ammonia emissions, and fertilizer application is the root cause of all NR emissions, NR losses due to processes like volatilization, denitrification, and leaching and runoff. So this is the total NR released into the environment from the cropland system. Here we can see that maximum NR is released in the form of ammonia, followed by non-reactive form, that is N2, and then NOx and N2O. So cropland causes maximum release of ammonia as compared to other NR, and it is 88% of the total NR released into the environment. Talking about the trade of 3Fs versus NR emissions, the 3Fs is like food, feed, and fuel. because the environmental dilemma is to establish the nexus between food, feed, and fuel, and as well as take care of the NR emissions. So here is the total nitrogen uptake by the crop is divided into two parts, grains and straws. Now, grain is taking 54% of the total NR accumulated in the plant, and then straw has 46%. This is the overall temporal analysis of the various fate of this grain, like human grain over the 2 decades, 2000 to 2020. And these are fates are human grain consumption, grain export, fish feed, livestock, grain feed. The different fates of straw are livestock, straw feed, domestic fuel production, thatching, other fates, burned on field, etc. And we can say that the trend of grain and straw fates open a room for policymakers to decode the food, feed, and fuel security issues with simultaneous reduction in NR emissions. So on summary and conclusion, NR is a multi-pollutant, multi-scale, multimedia challenge, and 60% of the NR enters in the hydrosphere and 40% has a fate in atmosphere, in which 35% is only ammonia. And ammonia based, because of its high spatio-temporal dynamics, needs special focus and needs to be studied in fine detail in future. Also, India's significant yield gap raises a compelling concern, while efforts to enhance food security are crucial. They inadvertently contribute to environmental challenge by adding excess nitrogen, impacting nitrogen use efficiency, and leading to release NR, intensifying the environmental dilemma. The integrated inclusion of atmospheric, hydrosphere, and terrestrial addition of NR is not reflected in today's regulation. So this study is the first attempt in India that studies the nitrogen flows to characterize the losses and impacts that have implications for nexus of 3F, that is food, feed, and fuel. And this study opens a room for policymakers to decode the food, feed, and fuel security issues with reduction in NR. And the sectors, some unregulated sectors like fertilizer application is the biggest source of NR release, and agriculture practices like fertilizer application and manure usage due to the volatilization process releases ammonia along with the deposition and needs to be considered to ensure food and environmental security. I would like to acknowledge the PMRF grant, that is, for those who don't know, it's Prime Minister Research Fellowship Grant, and DST and Professor my lab group CARES and Professor Chandra and Professor Harish Fuleria from IIT Bombay to help me with data finding. Thank you.
Any questions? The first question is related to the NGO emissions. Do you consider the denitrification or identification both? And how your emission from the agriculture sector of nitrous oxide compare with what India is reporting to the national body? And first question is that. The second point is, when you're combining the emission factors, are you taking global emission factors and you're taking the national emission?
So the first question, I am only considering denitrification processes, not nitrification. And other than that, comparison with the other studies. So yes, I have compared with the nationally reported data set like EDGAR and GFED. And EDGAR is like, so my emissions are underestimated as compared to EDGAR emissions, and other studies I think also shown the same pattern, like EDGAR emissions are 35 to 45% higher for other studies. So it's a similar pattern I am also getting. And what was your next question? Emission factor, yes. Emission factors for particularly N2, I'm focusing on national emission factors, Indian emission factors.
Because EDGAR is not a national report, I am talking about the national communication which India is spending to policy. How your emissions, like especially nitrous oxide emissions, are compared with that?
I have not compared it with that as yet, so I'll look into it. Thank you.
Question?
Just a continuation to what ma'am has explained, you have projected some values for the gaseous emission, identification. So, were the emission factors taken from a global literature or specific to the India?
No.
You have validated.
So for the major responsible processes, I would say like volatilization and fertilizer application, I'm considering only Indian specific emission factors. For those for which I didn't get like human excretion or something. I am considering other literature as well. So for denitrification, if you are asking, I'm accounting for the Indian emission factors.
Actually, like, Indian specific emissions.
Yes,
It is from the inflation.
yes.
If nobody has, then I want to ask. You showed, Lok Sabha questions,
Yes,
you
sir.
have included,
Yes,
about teaching ways.
yes, sir.
What was those two questions?
So actually I was looking for the kitchen waste generated from the, so there's an assumption that I have divided the whole population into two subsectors like urban population and rural population. I'm assuming urban population kitchen waste is not directly going into the croplands. So only rural waste is coming directly to the croplands. From that rural waste, I've looked into various questions imposed through Lok Sabha, and they have given this answers. Okay, from the rural population, this much of kitchen waste or organic waste is directly coming to the cropland system. So based on that, I have developed these numbers.
Which year was this question asked?
I can share.
That you can share.
Yeah, it's not that much old. I think 2015, 16 something. Yeah.
Now, I know another observation was regarding nitrous oxide. Your nitrous oxide is almost half of the deposition, through BNF and deposition.
Yes, sir.
So, don't you think deposition will be high?
Uh,
0.0306.
This one, sir.
Yeah.
N2 emissions is 0.12 and BNF. So comparing with the biological fixation, because biological fixation is not able to meet the food security demands of the rising population. That's why we are using this high fertilizer application and all. Usage of fertilizer is high. So if we compare with the biological fixation, this is fertilizer is twice releasing the twice N2 emissions as compared to the BNF. And deposition is half of that. It
It's too high.
It should be less than,
less than BNF. That's what you are.
very less.
Okay. Okay, I will look into it.
And also, which deposition you are considering, dry or wet or total?
No, I'm considering total deposition, not pollutant specific.
Okay, so.
Followed by deposition, are you considering the mineralization for the emissions?
Remineralization of whatever has been introduced from various inputs, that I am considering. But if you are asking like what is emitted and out of that what fraction is redeposited, that I'm not considering. Yes, I have come up with some number, because without considering the chemical transport of the pollutant, I don't think it should be included in the cycle.
Good, thank you, Deepakji, very nice. And very, yeah. Made very systematic flow chart, detailed. So now we have next speaker, Lumini Mofalirwa, who will speak on lichen responses to atmospheric ammonia concentration in case of gland pores. Lumini, please. 10 minutes plus 2.
So you can either use the other piece or use the space bar.
Thank you. Yeah, so the topic of my presentation is lichen responses to atmospheric ammonia concentration, a case of Glencos in Scotland. So to give an introduction of my presentation, I just want to highlight a few points to show why studying lichen responses to atmospheric ammonia concentration is important. So lichens are symbiotic organisms comprising a fungus and a photosynthetic alga or cyanobacteria. and the epiphytic lichens, that is lichens that grow on trees, are important forest ecosystem components and also bioindicators of air pollution such as excess nitrogen. However, lichens are highly diverse with contrasting sensitivity to increasing nitrogen levels. For example, excess nitrogen can negatively impact lichens, leading to species decline, but it can also promote the growth of specific nitrophytic lichen species. So in our work, we conducted field studies to elucidate the physiological and genetic responses of the sensitive and the tolerant epiphytic nitrates to enhance atmospheric ammonium concentrations, as well as to determine the critical nitrogen loads in lichen tissues. Our experimental site was at the Glencross Forest, which is near Edinburgh in Scotland. This is the experimental plot where we conducted where nitrogen is released, as you can see on the pipe there in the picture there, towards the direction of the wind, creating a gradient of atmospheric nitrogen concentration that decreases as you go further away from the release point of the ammonium. So I should mention here that the next presenter will explain more about this experimental plot. So for this work that I'm going to present, we transplanted lichens within the plot along that gradient of atmospheric ammonium at the distances of 5 meters, which is closest to the release point and has the highest concentration, and then at 13 meters and 32 meters, plus an additional control location that is not affected by the released ammonia. So those transplant locations, 5 meters, 13 meters, 32 meters, and the control location correspond to the atmospheric ammonia levels of 0.5, 12, 5, and 0.5 microgram per cubic meter atmospheric ammonia concentration. As I said, we conducted a series of experiments. The first experiment, we used the lichen species Evania prunastri. And on the pictures there, the picture to the left, showing where we collected the lichen specimens from the Scottish Highlands. So you can see the lichens growing on trees there in the forest where we collected the specimens. And then we took those specimens to the laboratory where we did baseline measurements and prepared the lichens ready for transport by fixing each specimen on a net. As you can see on the picture there, then the transplanting was done by, as you can see on the picture on the right there, by fixing that net with lichens on the trunks of the trees at those distances that I've explained. And our sampling points included time zero, which corresponds to the period in the lab where we did the baseline measurements, and then at 1 week after transplanting, at 2 weeks, and so on. And our lichen data collection included lichen growth rate, which is a measure of the whole lichen organism vitality, and also lichen tissue nitrogen status, and also electrical conductivity, which gives a measure of the fungal cell membrane integrity. I should explain here that the electrical conductivity is measured by taking a lichen sample and immersing it in deionized water for 1 hour, and then you measure the electrical conductivity of the water, so that it gives a measure of the leakage of ions from lichen cells. Or I should say a measure of the vitality of the integrity of cell membranes. And also we measured photobiont chlorophyll content. So just to show you some results from this experiment. So the x-axis shows the duration of exposure that corresponds to those sampling points that I mentioned. And we can see that the relative growth rate varied with the exposure duration, and that is the atmospheric ammonia concentration, and it was highest at the highest atmospheric ammonia concentration, that is at 5 meter distance. And that is from at least 2 weeks, from around 2 weeks of exposure duration. And the figure on the right there showing total chlorophyll content. And we can see that it also varied with the exposure duration and atmospheric ammonia concentration. And it was highest at the medium atmospheric ammonia concentrations, especially after about 1 month. And also electroconductivity, we can also see there that the figure on the left and also total nitrogen content of the lichens. The figure on the left there, we can see that both varied with the exposure duration as well as the ammonia concentration. And the electroconductivity was highest at the highest atmospheric ammonia concentration. That is both of them. And and also the differences, as you can see there, correspond to the levels of ammonia concentration lowest at the control point, which had the lowest atmospheric ammonia concentration. So we looked at the correlation analysis of these parameters that we measured. So we can see that the relative growth rate, which is the measure of the vitality of the whole organism, was strongly associated with the electroconductivity. Showing a negative association, a negative relationship with R value of 0.63, which is a value that shows a strong relationship. And this relationship can be seen in more detail in the figure on the right there. So what this shows that the lichen was shown to, that is electroconductivity was shown to be an effective measure of the vitality of the lichen organisms exposed to high concentrations of atmospheric ammonia. In the second experiment, we used 2 lichen species, that the Bryolia fasces, which is known to be sensitive to excess nitrogen, and the Xanthoria parentina, which is known to be tolerant. And in addition to the data that we did for the first experiment, here we are also looking at the lichen gene expression using transcriptome analysis, so that we can understand the genetic controls of lichen responses to excess nitrogen. And just to briefly examine results for this experiment, you can see that the total chlorophyll content on the above figure there was also highest at the medium atmospheric ammonia concentration, and also electroconductivity was also highest at the highest atmospheric ammonia concentration. And also that is also varied with the exposure duration. So this picture is just to show you the further experiments that we conducted that have recently completed, but I will not go into details of this. So in summary, our preliminary data show that the medium atmospheric ammonia concentrations may increase chlorophyll content in lichens, and the electroconductivity revealed strong responses of Evania prunacea and the Bryolia fasces to higher atmospheric ammonia levels, which thereby revealing possible fungal cellular damage. And also lichen growth rate was negatively impacted at the highest atmospheric ammonia concentration from as early as 2 weeks of exposure duration. And correlation analysis revealed that electroconductivity is the sensitive measure of the impacts on a lichen growth rate or the vitality of the whole organism, and thus it could be a useful, easy to measure area parameter in lichen biomonitoring of excess nitrogen, thereby revealing ammonia risk before lichen death. So these results elucidate important lichen responses that could support policy development for biomonitoring of nitrogen pollution. Thank you.
Now, open the question. Anybody? Any question?
Yes.
Thank you for this nice presentation. We are also working with lichens, or we have been working with lichens in Germany and trying to curve lichens from the natural surrounding into greenhouse. So this was very difficult. Lichens are very sensitive organisms. Did you observe how this transfer from the mountain area to this experimental site would influence the organisms?
That's a good question. Thank you for that. And it's interesting to learn that you are doing similar work. So I think that the key issue is how you handle the lichens from correction from the source until you transplant them, more especially the storage process. So we had we also considered this factor that you have mentioned, and we had a discussion as a research team that is with the researchers that listed there. So we discussed and we came up with the procedures for limiting that effect, which can lead to, for example, lichens responding negatively due to the shock of the differences in the environment. So we try, after collecting the lichens, we also limited the period of transplanting so that it's as short as possible. The samples were stored under that is cold conditions where the transplanting was going to take longer than 2 weeks after collection. So not chilled, but just cold conditions. So just to avoid possible death of lichens as well. So yeah, that's a critical point to consider. So that's what I.
Thank you for this. Any other place, one of the samples in the natural surrounding, but attached to this artificial substrate, and see how this then develop and compare it.
Yeah, so I was going to go to this slide. I think it's the other species, the Bryonia. So for the growth rate, okay, I've not shown that because for this species I showed the selected data, but for growth rate at the most sensitive species, that is Bryonia, at 1 week, the growth rate went down. So which was probably because of that transplanting shock, because of the difference in the environment. So which is the issue probably that you're trying to, yeah. But,
Any other question? Not then, I just want to clarify one thing, please. You were talking about chlorophyll total or chlorophyll A and B?
so we looked at chlorophyll, total chlorophyll content. I should have mentioned that because here we are looking where the parameters that we selected are parameters that are commonly used for biomonitoring, and the other thing that we're looking at is try to identify the most effective parameters that can be used at large scale that are easy to use. So most of the chlorophyll measurements, so most of the chlorophyll measurements involve extraction methods which are difficult to implement at large scale. But what we use here is a simple instrument that is called CCM 300. So that is, so it may say you.
Related to that, you say that exposure of ammonia enhances chlorophyll production. You know? Yeah, and but 25 microgram and above, that impacts negatively. What are those impacts? In fact, what happens?
Yeah, so that's something that the results that we also discussed. We're also still discussing as we're writing up this work. So we also have to go deeper into that. So as I've said, the chlorophyll was higher at the medium concentrations. So possibly the highest concentration was negatively impacted by the lichens. But that is had negative impacts on the lichens. That's why we did see we found lower chlorophyll content at the highest concentration.
The optimum range beyond that it is.
Yeah, probably there's a optimum range. So the other thing that we'll do is when we put all the data together, try to find the optimum that is logged.
Very good. Thank you very much.
Thank you.
Yes.
So I'm inviting our third speaker, Ajinkya Deshpande, please. And sorry for pronunciation. You will correct that.
That was correct.
Okay, thank you very much. So in 10 minutes. you have-
10 minutes will be the first thing. Then I will.
Yeah, and then only 3 minutes for questions. if there's some-
I'll try to keep it up.
Okay.
Using this, otherwise this always will. So that's your position.
I think so. It's not going to show on the screen. That's fine.
Yeah, the whole thing.
So thank you for showing up to this presentation. So this work is basically the first part of what Lumbani just presented. So this should have been the other way around. I should have gone first, but that's fine. So I'm going to talk about what these experiments are, how they have been set up, and the results from estimation of ammonia deposition onto different surfaces within the forest canopy. And we'll be doing that using a resistance model. So I would like to start by mentioning why we focus on ammonia. So ammonia, as we know, is a silent ecosystem killer. This has been talked about several times since yesterday. But UKCEH has been working on this for the past 30 years, and we've seen a wide range of impacts that ammonia can have. That first picture that you can see up there, that is our site at Wimbog, which was established about 25 years ago. And you can see the circled area, that's the higher concentrations of ammonia. The native Calluna vulgaris, which forms the blanket bog, has been completely replaced by Eriophorum grasses. So we see a complete species change under high ammonia concentrations there. Other than that, we see mosses and bryophytes getting affected. So you can see that lower picture over there, moss completely turns to slime at even moderate concentrations, not even high concentrations of ammonia. We've seen lichens getting affected that Lumbani just talked about, and we have one more presentation after this, so you'll see more effects on lichens. And we use lichens as bioindicators exclusively because they get all of their nutrients from the air. So they are perfect bioindicators for air pollution. And so when we don't have extensive monitoring, we focus on lichens, and they give us a good indication of what the air quality is. It also leads to vegetation damage, large-scale mortality of plants, imbalance in soil chemistry and plant uptake. We are also working on looking at the emissions of N2O from soils under the effect of high concentrations of ammonia. And that basically happens because of the imbalance in microbial activity in the soil. So ammonia has a very overarching damaging impact on the ecosystem, and therefore we like to call it the silent ecosystem killer. Just a quick recap on why this is important. We've seen these maps and plots several times across different talks since yesterday. So South Asia and Africa, parts of Africa, have emerged as massive ammonia hotspots in the last few years. And therefore, we are focusing in this region. And we think if we focus on South Asia, we can use this as an example for the rest of the world to tackle ammonia pollution. The bottom plot there shows that the emissions of ammonia are not going to reduce for the next few years, and the problem is going to get worse, at least until 2030. And so there is an immediate need to tackle this. The problem is that there's no real policy in place to tackle ammonia pollution in South Asia, especially in terms of its effects on natural ecosystems. So I would like to talk a little bit about ammonia critical levels and the critical loads of total nitrogen. So a critical level is the concentration of ammonia in the air at which a given organism starts showing negative impacts. And the critical load is similarly the deposition rate of total reactive nitrogen beyond which a given organism starts showing negative impacts. In the UK and EU, back in about 2006, 2007, critical levels and critical loads were established. And these were then adopted into national policies across Europe, which means that whenever there's a new project coming up, an infrastructural project like a highway coming up, or a poultry farm, you have to prove that in the vicinity of your project, the ammonia concentrations will not exceed critical levels, and only then you get a permission to set up a new project. So these kinds of policies and regulations are required in the South Asian region, and that's what we are trying to facilitate using these experiments. So UKC had set up an experiment at Wimbog in 2001, which was instrumental in forming the critical levels and loads in Europe around 2006, 2007. However, it's not just important to determine these critical levels and loads, but the timely revision of these levels is also important because they change over time as organisms get exposed to pollutants for a longer period of time. So the objectives of our study is the controlled enhancement of ammonia air concentrations in forests under field conditions to replicate real world pollution scenarios. So we try to enhance the concentrations to a level that is more realistic. There have been experiments in the past, but other researchers have enhanced the levels to extremely high levels, which are not commonly seen. So we try to keep it realistic, and we try to replicate conditions that are observed near ammonia point sources. Secondly, we want to understand and quantify how this gas behaves within a forest canopy, how it transports, how it moves, where it goes, where does it deposit, and therefore identify the canopy layers and the organisms within the forest ecosystems that are at maximum risk of getting exposed to high ammonia concentrations. And lastly, we want to study the impacts of high ammonia concentrations and deposition on vegetation to determine critical levels and loads. This work is mainly spearheaded by Lumbani and Buddhika will be showing his work later in this session. So we established two very contrasting study sites, and establishing these contrasting study sites gives us the opportunity to test our experiment in a very wide range of conditions. So our first site was established just south of Edinburgh, in a birch plantation forest with a very sparse canopy, as you can see in the picture. The forest is deciduous, loses all its leaves from about November up until March. So for 5 months, we don't have any leafy surfaces for ammonia deposition. The conditions are, as expected, cold and humid, as you would expect in Scotland. In Sri Lanka, we have a site in the tropical submontane forest in the central highlands of Sri Lanka at an elevation of 1,400 meters, 1,600 meters, with a very dense canopy. You can see the difference between the canopy structure at the two sites. It's very sparse and very layered in the birch plantation, whereas it's more uniform and dense in the tropical forest. And it's an evergreen forest, so we get leaves all throughout the year for deposition. So a quick look at our method. So what we have done is we've set up a meteorological tower, which extends beyond the canopy. We measure all meteorological parameters all the way from the ground level to the top of the canopy and beyond. This tower also has an ammonia cylinder attached to it at the base. We measure wind conditions at about 2 meters from the ground, and only when the wind conditions are correct, which means that the wind is blowing towards our study transect, there's a valve that opens and it releases ammonia through the pipes towards our study transect. So only the part that we want to get exposed to ammonia, it gets exposed. So it's a very controlled experiment. And downwind in our transect, we have put up alpha samplers. So those who are not aware, alpha sampler is a small passive sampler for ammonia, and it gives a monthly average concentration. So we've literally packed the whole site with alpha samplers so that we can track where the gas is going. From time to time, we also set up a high resolution Picaro analyzer to look at the fine scale movement of the gas, where it's going, if it's where it's getting transported. And then we use a resistance model. So I will not get too much into details of this because it's really time consuming, but the paper has been just published and I've put a QR code at the end of my presentation. So if you're interested, scan the code, read the paper. But basically, ammonia deposition is estimated using the resistance analogy, which means on the left side, these are concentrations of ammonia in the air across the canopy layers, and on the right side are the surfaces on which the gas deposits. So on the x-axis, you have the distance going away from the source. The gray line is the Sri Lankan side, and the black line is the UK side. The Sri Lankan side has bidirectional release, so it releases on two sides because it's a larger plot. And you can see the concentrations, it goes up to 55 micrograms. So we've been able to enhance it to quite realistic levels. And we've been looking at vertical dispersion of the released gas. And we see that in the sparse birch plantation, the gas likes to settle down close to the ground. And so the ground flora and the bryophytes get exposed to quite high concentrations. But in the Sri Lankan forest, in the more denser canopy, because we think it's because of the better mixing of the eddies, the gas then gets transported upwards. So we have identified 6 different pathways where the gas can deposit in the forest canopy. So the soil surface, leaf cuticles, stomata, the trunk surfaces, and for vegetation, we have the ground flora layer and the tree canopy layer. For the UK side, we can see that more than 1/3 of the gas deposits onto the soil surfaces. So that's a massive sink for ammonia. And more than half of the ammonia deposits onto the leaf cuticles in the ground flora layer. Whereas at the Sri Lankan side, we get 1/3 of the ammonia depositing on the soil surface and about 1/3 on the leaf cuticles in the tree canopy. So the gas does go upwards vertically. In terms of larger implications, we found that about 26% and 64% of the released ammonia gets recaptured within the given plot. So it shows that these forests have quite high recapture potential of ammonia, but that also means that they get, so if you have a forest really close to a point source, it can get exposed to really, really high concentrations and deposition levels. And secondly, we want to provide these new deposition velocities that can improve large-scale modeling efforts. So just in conclusion, we've been able to reproduce real-world conditions of ammonia pollution, and we're getting some first insights on how this gas behaves within forest canopies. We've identified the layers within the canopy that are most at risk of ammonia deposition. And the resistance model is new because the work that has been done so far, it has been done on looking at ammonia deposition from top to bottom, so above the canopy from the atmosphere onto the canopy. Whereas in our case, we are releasing from below the canopy, so our gas moves from bottom to top. And that's the new part, because then you can look at forests that are really close to point sources, and you can see how the gas would behave in these patches of forests that are really close to pollution sources. So that's me, and that's the QR code. If you want to read the full paper, it's open access, so feel free to take a picture. Thank you so much.
Yeah, thank you for that nice, thank you for the nice presentation. I think, yeah, the point you mentioned at the very end, I think is very important. So we think that the results can really apply for when you have like the source and it kind of penetrates
laterally through the trunk space.
So,
Yeah.
And the position loss is towards it, because you say like they shouldn't be applied then, they shouldn't be applied for that case then, right? If you have like a vertical or horizontal transport into the canopy, not on the top.
Yeah.
Right?
So I'm sorry, I don't know if I completely understood your question, but the deposition velocities that I presented is the total, like the equivalent total deposition velocity for all the canopy layers, but we do have individual deposition velocities for each layer as well, which I think they are in the supplementary material in the paper. So
But then you don't come to the top, those may be different.
no, we don't consider RA, which is the aerodynamic resistance. We've completely removed that from the model.
May not get to the like into the kind of the problem, right? So I'm wondering, because you have like, I guess you have like parameterizations maybe you can drive to then later back calculate how it would be like when it's coming from the top.
Yeah, we can do that. And we also measure background levels. So we know in addition to what we are releasing, what's coming from top, from above the canopy, we have measurements of that as well. So yes, we can do that.
I have a question. Yeah, sorry. When you said there is difference in the soil, deposition on the soil between the two parts.
Yeah.
Do you know why? Is there a reason of the soil structure, for example?
Yes, and so this is a part of a PhD project under our project. But I think it has to do with the existing ammonium concentration in the soil. Yeah, so if you have already high concentration of ammonium, you would get a lower deposition onto this. So I think that's the biggest factor.
The last question. How come we?
Could you expand a little bit on how the long has been released from this tube that you showed there on the program?
Yeah, so what we have is a mass flow controller connected to a data logger, and the data logger is pre-programmed in a way that we have 3 wind ranges, and at each wind range, we have told the data logger how much of the mass flow controller should open, and the mass flow controller valve then opens, it turns on a solenoid valve, and that's how the gas gets. So we have a fan at the other end which pulls the gas through, if that makes sense.
The last question.
Very good presentation. So then you said that 52% is deposited on the soil.
Yeah.
So is there again volatilization and backpress ammonia? I mean, is there a possibility volatilization back to the atmosphere, and also are you monitoring any sort of a soil mechanical or properties in this situation?
So I'll answer the second question first. Unfortunately not, because Galena could not go back to Sri Lanka. So we're not able to follow up on that. The first question is yes, the model does account for re-emission. So if you look at the monthly deposition results, during some months you get re-emission instead of deposition. So yes, the model does account for that.
Yeah, that's one question regarding Sri Lanka, we have very efficient deposition instrument, almost double of the one.
Yes, there's a very clear reason for that. That is because we have more sunlight in Sri Lanka for a longer period. Days are longer, summers are longer, so the stomata remain open for a longer time. And so it's open, so it can take more ammonia in as compared to the temperate forest.
Yeah.
Thank you.
Thank you very much. We have now presentation by Mala Pokhriyal on simulation of nitrogen deposition in the Indo-Gangetic plain using WRK model. Mala, please.
Okay, thank you
And
everyone
plus
for
two.
coming. Okay, today my topic is simulation of nitrogen deposition in the Indo-Gangetic plain using the WRK model. This is my research presentation outline. Coming to research background, nitrogen is an important
part of protein, nucleic acid, and chlorophyll. It can be found in various form, as you can see from this figure. It can be found in gaseous form, aerosol form, and aqueous form. And but if it is deposited in high amount, it can lead to eutrophication, soil acidification, and poor air quality. Talking about the nitrogen deposition in the intergenetic plane, as you can see from this figure, numerous studies have consistently shown that nitrogen deposition is very high in South Asia, and within the South Asia too, in the IGP region has been emerged as a global hotspot of nitrogen deposition. So it is very important to understand the nitrogen deposition in IGP. Now, these are my research objectives. This includes assessment of model performance to simulate different forms of nitrogen, quantification of nitrogen budget in the IGP region, and to find the key drivers of nitrogen losses across the IGP. And this is our study domain. It consists of a Corsair 90, it is a nested grid consisting of a Corsair 90 kilometer grid, and within it there is a 18 kilometer grid. Our model has 33 vertical level, and our simulation period is only, well, currently it's only Jan 2018. These are the modeling setup. We used WAV-K version 4.2.2. The initial and final boundary condition were from ERA 5 data with a 3-hour interval. For gas chemistry and aerosol, we used Mozart-4 mechanism. For chemical boundary, we use CAMChem in CSM2. For emission inventory, we use Edgar version 5, and biomass burning inventory, we use the FIM version 1.5. Biogenic emission was from Megan version 2.1 plus, and for dust, we use the GoCart. These are further setup. We use the default scheme for dry deposition in cloud scavenging and below cloud scavenging within the model. And for validation of data, for air pollution, we use most of the data are from CPCB. In Nepal, for Nepal, most of the data are from Department of Environment, and we also used US Embassy dataset. The meteorological data were downloaded from CPCB website, and other South Asian sites were downloaded from Mesonet. And wet deposition data are from provided by Professor UC Kulseshwar sir and Dr. Yogendra Singh. Now, talking about the performance, these are the model versus station PM10 results in Nepal. The model had a moderate correlation in Dhulikhel region, which is a semi-urban site in Pokhara and Kathmandu. However, the model had a very weak correlation in Lumbini. In India, it had a moderate correlation in all sites except in Noida, and model underestimated the PM10 site in all sites except Burari. Okay, these are for the data from India. The data had a moderate correlation in all sites except the same in Noida, and the model underestimated PM 2.5 emission in Noida, while it underestimated in other regions. Okay, these are the weight deposition results for January 2018. The model, these are the differences between model and actual precipitation data. And for nitrate, the model overestimated in all stations, while for ammonium and the model underestimated in these stations. Okay, these are the weight deposition results. As you can see from this figure, between 72 to 80 degree and 25 to 30 degree, ammonium had high flux of around 30 milligram of ammonium per square meter, whereas nitrate ion had a flux of greater than 40 microgram of nitrate ion per square meter. Okay, these are some of the dry deposition results from the model. As you can see, the southeast plane of IGP has concentration flux of 4 to 6 milligram of ammonium per square meter. In other region, it is only about 2 to 4 milligram of ammonium ion per square meter. In southeast plane of IGP, it is around nitrate is around 12 to 18 milligram of nitrate per square meter, while in other region it is 4 to 6 milligram of nitrate ion per square meter. Okay, these are my key findings. The model showed moderate correlation between PM10 and PM2.5 for most region. However, there is for in wet deposition there was underestimation in all available station. While for nitrate ion, the model overestimated in all region. Now, there was a significant wet deposition of observed between the 72 to 80 degree east and 25 to 30 degree north with ammonium concentration of 30 milligram of ammonium per meter square and nitrate of greater than 40 milligram. Sorry, ammonium of 40 milligram per meter square. And for aerosol, ammonium ranged from 4 to 6 milligram per square meter, and nitrate varied between 12 to 18 milligram per square meter. The wet deposition flux in other regions were comparatively lower. Okay, I'd like to acknowledge UCRI, CCRF, South Asian Nitrogen Hub for funding this project. Now, these are the references. Thank you.
Thank you very much, Mara. Now, anybody want to?
Hello, I was quite impressed with the difference in the PM and PM and the model. I can imagine that I'm from Portugal, so our agreement is completely different, but the magnitude is also. Well, maybe this relative error is more or less centered, even like that. I think your moderate agreement, it's a little bit optimistic. Okay. But my question is, do you think this underestimation of the position that you mentioned of the ammonia could be also related to the big underestimation that you had for the concentrations of PM? Because you mentioned just the numbers, but you didn't suggest any explanation for that. So that's why I'm wondering, what kind of explanation are you suggesting for your results?
Actually, my results had come just a few weeks ago before this presentation, so we still need to analyze the data. Yeah, so when I will further analyze, I will tell you.
Okay, yeah. Thank you.
Yeah, you have to see after going back home.
Yeah.
You see the 3 points.
Yeah, okay.
CLA and one already it is there in your conclusion. The other one I'm adding that underestimation of ammonium and overestimation of nitrate. What are the factors? Whether it's the modeling parameter or with the chemical analysis or field work.
Yeah, okay. I will have a look.
Okay.
Okay.
Thank you, Mala.
Thank you very much.
Next speaker is Budika Wirakul about the forest in Sri Lanka. Yeah, okay. So you have 10 minutes and 5 questions. Thank you. Yeah.
Thank you. So let me start my presentation. Topic on Yeah. Yes, thank you. So the topic of my presentation is responses of submontane forest ecosystems in Sri Lanka to increased atmospheric ammonia concentrations. So my name is Buddhika Veerakon, and this study is co-authored by Matthew Jones, Ajinkya Deshpande, Chris Ellis, Gautami Veerakon, Sarat Nisankar, and Mark Sutton. So let me go through a brief introduction about the study. So as you are aware, since yesterday, so ammonia is one of the main reactive nitrogen atmospheric compound, and agriculture is the one of the main sources for atmospheric ammonia. So and also you have noticed that there are several impacts of the excess ammonia to the environment and also the human health. So environmental impacts that may be like, so acidification of the soil and biodiversity loss, and also the biodiversity loss, sorry. And for the considering the human impact health, it's you can see that the respiratory illnesses are one of the main human impact to the from the atmospheric ammonia. So but even we know the atmospheric ammonia can cause damages to the environment, but we have to quantify it. So the main objective of this study is to assess the responses of forest ecosystem to the enhanced ammonia concentrations. To achieve this objective, we have established a forest plot in the Rilagala forest, which is located at the tropical submontane forest in Sri Lanka at the 1650-meter height elevation in the highlands of Sri Lanka. And this forest patch is surrounded by tea plantations, which is one of the main agricultural practices in the Central Highlands. And also this forest, the forest plot is the first forest plot this kind of established in the tropical range of the world. And let me go through a small introduction to the forest. So the plot is 120 meter and 40 meter by a plot, and we have divided it into 192 subplots, which is 5 meter by 5 meter. And we have a ammonia emission system at the middle of the forest plot, so 20 meter in long. So we are monitoring bidirectional ammonia fumigation there. So 30 meter into the east side. So we have a 60 meter by 20 meter area we are monitoring continuously during the study. And as Ajinkya explained earlier, we have a meteorological tower with the fumigation system at the middle of the forest plot, and we have several alpha samplers there to measure the monthly ammonia concentration in each locations. So, and apart from the ammonia concentration, we are monitoring the impact to the forest vegetation at that plot. So we are monitoring the bark pH and also the lichens. So lichens, we are monitoring the temporal variation of thallus area of each lichen. So let me let me take you to how we are monitoring the lichens. So we are selected the lichens species that for lichen thalli that from the ground level to 2.5 meter in height, and we have selected them for long-term monitoring. How we are doing the thallus area monitoring is we have prepared divided into 1 square centimeter, and we printed it on a transparent sheet, and we put it on the lichen thalli and count the area. And we are measuring the thallus area continuously from April 2022. Let me go through some results we got from the study. So we have identified 31 lichen species in the sampling plot, and also we have identified several lichen species have dominated the plot. Here you can see Leptogium sinensis, Cococarpia pellita, and Xenogonium blinky. Those are the most abundant lichen species in our forest plot. And as I mentioned, we are monitoring the temporal variation of the thallus area. So we have seen much negative responses from these species than the other species. So what are the negative responses we saw? So we have noticed that the thalli color change is changed, and also thalli decay, and also disfragmented, and ultimately some thalli are eradicated from the plot. So those are the negative impacts we have observed. Here you can see in the first picture, a heterodermia species thalli has changed its color, it bleached. And in the second picture, you can see the Xenogonium blinky thalli it's going to fall from the attachment from the bark. And in the third picture, it's the Leptogium species, and it's decaying. And at the final picture, the fourth one, it's a Sticta species lichen, and it's already dead. Sorry. And we have put some time-lapse cameras in our plot. So we are monitoring those. We have put some four time-lapse cameras, and we have focused them into 4 selected lichen species. There are 2 cyanobacterial lichen species and 2 algal lichen species. So we are taking 1 picture per day in each camera to measure the temporal variation of the change of the thallus. So here you can see some changes. So the first picture is taken in the 2022, and the second picture 2023. Here you can see how the thallus have changed with the time with the impact of ammonia concentration. And I have prepared some graphs for you. So in the first graph, this is some selected lichen thalli of the Xenogonium blinky species. Here you can see the lichen thalli very close to the fumigation system had shown early responses than the lichen thalli located away from the fumigation system. So most of some lichen thalli, they have reduced, they have eradicated from the system, but there are some still surviving lichen thalli. You can see there are some still surviving. And the second graph, it's for the Cococarpia, and third graph, it's for the Leptogium. For all 3 graphs, for all 3 species, you can see that the lichen thalli close to the fumigation system, they have shown early responses than the lichen thalli away from the fumigation system. Apart from the lichen thalli, we have measured the bark pH. The bark pH, when comparing the bark pH with the before fumigation and after 1 year fumigation, they have shown a clear variation there. So what we have recorded is bark pH has increased. So bark pH increased with the fumigation, and the trees close to the fumigation system, the pH change is higher than to the trees far away from the fumigation system. And moving towards to the discussion of my results. So we have identified that high ammonia concentrations are affecting the lichen thalli, and based on those information, we can identify acidophilic and the nitrophilic lichen species. That means which are preferring the acidic conditions and which are preferring the basic conditions. And based on those information, we can identify that which lichen thalli, which lichen species that can be used as bioindicators for ammonia pollution. And finally, this information can be used for future policy making and mitigating the negative impacts of ammonia pollution. So these are some references, and thank you very much. Yeah.
Thank you very much for this very interesting talk. Just, I fully understand the negative effects, but we know with respect to the climate change effects of nitrogen. Very often it's said that the deposition of the nitrogen also supports more vigorous growth of forests. So we have a carbon capture. Do you see that things observed for your forests in nitrogen ahead?
Yeah, apart from these negative impacts, the negative observations, we have seen some positive impacts of some lichen species also. Some lichen species, they have increased their growth. So as I said, we have acidophilic and nitrophilic lichen species. So the species which prefer more basic conditions, they are nitrophilic, so they start growing. But at some point, they also maybe show some negative responses. So apart from the lichens, so yeah, apart from the lichens, we have observed some other vegetation responses also. So which we are not documented here. So we have observed some shrubs and undergrowth bushes. They have grown comparatively higher rate than the non-ammonia impacted areas. So yeah.
Yeah, thank you. Yeah, very interesting experiment. Thank you for those insights. Could you tell us about the concentrations that you measured, where you observed those changes in light and growth rates? What
Yes.
were the concentrations?
So here, the concentration is range. So the highest concentration we have recorded close to the fumigation system, it's around 50 micrograms per cube meter. And at the very end, the far away, it's reducing to almost to 1 micrograms per cube meter. So these values are average values for the period of time. So we have the system is bidirectional, so therefore the fumigation, it goes to both directions. And with the wind patterns in the monsoon pattern of the country, so one part of the plot get more concentration than the other part of the plot. So the high concentrated part, it show more negative impacts than the other part. So
this question is, when you showed the lichens dying, where exactly on your plot?
Yeah. Yes. Here you can see the lichen thalli very close to the fumigation system, 5 to 10 meters. They show very rapid responses. Here, within first 200 days, some lichen species, they have shown very negative responses at here. But the far away lichens, they start showing responses around 400 days. So close to the fumigation system, more rapid observations we can.
Very much.
So we have next talk by Alexander Mover on emissions and bidirectional exchange of ammonia. Ever we call Alex, please.
So where do I proceed with the slides? Proceed with Okay, perfect. Okay, thank you and welcome. I'm going to talk about the dynamics of ammonia bidirectional exchange and emissions in the example for a corn crop canopy. So just as a quick introduction, we've seen that before. So we know ammonia is being emitted into the atmosphere and plays an important role in atmospheric chemistry, leading to aerosol formation, thereby impacting climate and air quality. Also, we've seen in the just talks before, nitrogen deposits and leads to eutrophication of ecosystems. Therefore, it's critical for us to know the emissions of ammonia. And this is also something that has been talked about yesterday in a talk where we see that in this example that ammonia is a major source for fine particulate matter. Here it's shown that in some periods or regions of the world, it contributes up to 50%, or it's responsible for 50% of PM2.5. Also, if you look at deposition here, an example for Germany, because I currently work at the German Environment Agency, we see here that in Germany, the deposition has been slowly declining over the last few decades. And we see here a deposition of oxidized and reduced nitrogen species. However, we see that for ammonia, the green here, the dry ammonia deposition has not been decreasing. So that's also because emissions have not been decreasing as NOx emissions. So emissions haven't been decreasing. So yeah, therefore, it's important for us to know the underlying processes, what determine that dry deposition. And if you're interested in that time series, we have a poster out there that shows how the time series exceeds and also has a scenario for 2013. So what did we do? We measured ammonia fluxes over 2 growing seasons in Canada above a corn crop canopy. One period from May to October 2017 and the other period from May to October 2018. We used a micrometeorological approach. Therefore, you see here a flux site where we need a fairly large footprint. And our objective were first to quantify the ammonia emissions after fertilizer application and also study or investigate the action processes of ammonia within the corn crop canopy. How do we do that? We use the eddy covariance method. For those who are not familiar, the eddy covariance method is the most direct approach to measure ecosystem scale exchange fluxes. So we need a measure of the turbulence. Basically, a sonic average gives us vector of the vertical wind vector of the vertical, and we have the vertical wind speed, and that is coupled with a very fast chemical measurement. We used here a QC tilde as a laser absorption spectroscopy instrument that can measure so fast. For ammonia, this is especially challenging because ammonia is a very sticky molecule. It absorbs surfaces, desorbs. That's why these measurements haven't been done in the past. Just in the last 10 years, there are successful studies that have measured ammonia exchange with the eddy covariance method. And we've collected, and now I come to the results, 2 seasons of ammonia fluxes. It's a very unique data set because it hasn't been, there is no data set that has collected such long flux measurements with the eddy covariance method. And what you can see here, right at the first glance, that we have both periods of emission and deposition. And that kind of tells us already something about the bidirectional nature of ammonia exchange. So I first want to focus or zoom into that period of fertilizer application. We see here 2017, we see there are some emissions. 2018, we didn't see them, right? Of course, the question is why? But in this talk, I'm only focusing on 2017. So what has been done? A fertilizer was applied. It was a granulated urea. It had partially an inhibitor with it, and it was broadcast, applied, and incorporated, which is very important because that determines in the end how much is being emitted. And here on the right-hand side, you see the fluxes for that part where there was no inhibitor used. And we see that after virilization, it needed some rainfall, and we were lucky that occurred just a few days after virilization. It rained, and that rain leads to the hydrolysis of the urea, and thereby we can observe the release or the emission of ammonia. Then you see here, for example, on the second day, there's another small cloud here. It was a rainy day that impacts also the exchange, and we see here that there has been no emissions. Maybe yeah, here you see that here, right? And on third day, we see again some medium emissions, and then they kind of taper off to the baseline, right? And so if we look then at the cumulative emissions, we see that around 0.1% of the applied fertilizer has been emitted as ammonia into the atmosphere. So, and to put that into perspective, for example, in the EMAP guidebook, so it's from a European perspective here, emissions from urea fertilizer are given there as 60%. So it's 2 orders of magnitude different. Like there's another study here that has even, it's a Canadian study, that's why I also listed it, has even higher values, but there's also other studies that have very low values. Here's a 0.4, for example. So it's in the range what we have observed. So we see here that the range of emission factors is huge. And our study is another point in that kind of cloud of emission factors, saying that emissions can be under certain methodological conditions very low. Now I come to the second part of the my presentation, talking about what happens actually when the canopy is developed, because we observe bioreduction exchange, and these processes are important to understand. And you can see here, I'm focusing here now again on the year of 2017. We see here that there was an emission period in August, followed by a deposition period in September and October. And to investigate that, I used a 2-layer compensation point model. Thankfully, my speaker before already mentioned that resistance scheme, and I have now, I'm lucky now that I'm able to dive a little bit deeper into that. So we see here the fluxes in green, and they are determined by those compensation points. Those compensation points are basically concentrations within the canopy, and the difference between those concentrations determines the flux size and also the magnitude. But the magnitude is also governed by those resistances. And we have, for example, here we have one soil pathway, and basically the transport from the top through the canopy to the soil. And if we look at that, of course, we know that the corn is growing over the season, and I've listed here, or probably here at the top, the leaf area index. And we see it's increasing. It's a green leaf area index, therefore it's kind of decreasing towards the end of the campaign. But what we see at the bottom here, and if we just focus at those brown bars, that's the flux with that model, or what the model predicts. And we see that the soil flux is negligible when the canopy is grown. Why? Because there's no exchange, or the exchange is very limited. For this case, could be different for a sparser canopy, but for our case, what we saw that the exchange is limited. Then there is the third path, a second pathway, it's a stromal pathway. And for you to understand what that means, or what that is actually, so the flux is driven by the concentration difference, the ambient ammonia concentration, and the concentration in the substomal cavity. And how, you may wonder, how do we know the concentration in that substomal cavity? Well, we can model that by using these so-called emission potentials. And these are very important because I'm going to talk about them later again. So, and those emission potentials are basically coupled through the concept of the partitioning between aqueous phase and gas phase, and the Henry's law constant is used here. And then one can actually, if one knows the emission potential, one can derive the compensation point. And these emission potentials are very important because those are actually in the models. And the models we've also seen in the previous talks. And what we see here, that they are ranging over like a certain range, and they also have a large uncertainty. And you may wonder, oh, okay, is that now a big uncertainty or what does it matter? And I can tell you that already little changes, maybe 10, 20% in our model led to different outcomes, meaning that, for example, we changed suddenly from emission to deposition. So having those numbers accurate is very important. The third pathway is the non-standard pathway. It's a deposition to the cuticles. And let's say many models thought to be unidirectional, so ammonia deposits, basically, and it's a function of the relative humidity and the amount of acids and bases on the surface. And if we now use our flux data and see if we can observe that, we see here the nighttime fluxes, because at nighttime we assume that all the stomata are closed. Therefore, we only have this non-stomatal flux. And you see here in blue the fluxes for 2017 at night. And in red is the model. First, we can see that the model doesn't do such a good job, right? And the second thing is that we also see in August at nighttime, we see emissions from the cuticles. So that means that we have to go from like a unidirectional approach to a bidirectional approach. And that has also been suggested in past literature. That's a concept here where we actually use this cuticle emission potential as well. So there could be release from ammonia from the surfaces. And there's also like polarization that's been used in some atmospheric transport models. It's now, so I'm adding now another one. It's the orange points. And you can see it's a different results, but it's again, it's very different from what we measured. So that basically shows the uncertainty in the measurements, in the models, and also in our understanding of the processes. So now what did you want to do here? So we have measurements over a long period of time, and our goal was actually to retrieve a new polarization. So I have that here shown again. So we have the measured nighttime fluxes. From those, we can retrieve those nighttime critical emission potentials, and then ideally have a polarization of function that describes those emission potentials. And we can use those now from the night, transfer them to the daytime, and get the daytime critical emission potentials, and then basically with our flux measurements retrieve modeled ammonia fluxes. And as I said, those functions basically of the critical and thermal emission potentials, these are the ones actually we were targeting to get a better understanding from our flux measurements, because we have this nice data set. Well, and now actually, so this is actually the introduction of my talk, I have to say. So, and I think I'm running out of time. Yes, yeah. So that's why I thought I'll leave the rest to another discussion in the coffee breaks, basically, right? So actually what we saw, I just wanted to get to that you get the main understanding. Actually, so that transition from night to daytime, actually, we found that it's really difficult because we have all different orders of magnitude in the fluxes. Nighttime fluxes are very small and not necessarily a polarization that we have from nighttime, we can transfer to daytime. Also, we have another problem. We have collected too much data because we have like so many different situations, conditions that not one single polarization fits the wide range of conditions during the growing season. So previous studies have often used like smaller data sets, and we are lucky to have a larger data set, but we tried really a lot and not successfully at having like a nice new polarization, which was the goal, right? Therefore, the hypothesis that also our big leaf compensation point models might be somehow limited in representing the bidirectional exchange. Or maybe we also have to, what we didn't consider is re-emission from the canopy. We didn't make measurements in the canopy. There could be other processes we wouldn't have to look at in further studies. So I encourage you to then come later. If you're working on this, we can have an exchange on that topic later in the coffee break. So with this, I'd like to conclude that we observed very low ammonia emissions, aurotization below 0.1% of fertilizer emitted, applied was emitted. Also, because bidirectional change is such an important factor, we see that the ambient concentration, so people who do like chamber study or studies studying ammonia emissions, if those are very small, the ambient concentration needs to be also considered. when applying emission factors. We see that the nitrogen budget was mainly dominated by deposition in the both two growing seasons. And however, the ammonia input from the atmosphere was relatively small. I didn't show that, but it was relatively small compared to the nitrogen input from the volatilization. And on the binary exchange, as I just mentioned, there has been no single polarization that could have been found. And we still have to work in the whole community. We need more measurements as well to basically pin down better those polarizations. And with this, I'd like to thank you for attention. So working on this was me and also Soumya Singh, which is here in the audience. We both switched to different affiliations. That's why it's one of the reasons why we haven't published this work. You've seen it from 2018. It will be published because we have the technical paper out. It will be published, so stay tuned for that. Thank you.
How many are there?
I have one question. We are already.
Yes, please.
I was.
So I just wanted your opinion. So in our study, we saw that the cubicular deposition pathway was the more dominant one. But in your crops, the stomatal deposition was dominant. Do you have an idea why could that?
Yeah, so I showed one figure. You saw that, maybe others haven't seen that, where the cuticle was very small, right? And that was, I mean, that was with the Massad name it Sutton polarization. Maybe probably use it as well.
We use 4 different.
Yeah. So if I use another one, the picture completely changes, and that's kind of the crux of the whole thing. Yes, yeah, basically. So
Thank
Thank
you
you
very much. Discuss. Thank you. Next. So next speaker. Hello, welcome. So about nitrogen dioxide, we will change a bit of the subject. It's not about ammonia. From satellite measurements. Thanks. So.
Thanks.
Minutes.
10 minutes and 2 minutes. Okay, thanks. Hello, everyone. Shift to ammonia to a little observation and instruments. It is a pure modeling work, which I have done under the Commonwealth Rutherford Fellowship Program at Edinburgh University. So this is a modeling work in which I am evaluating a global climate model, particularly over the South and East Asia. So this is South and East Asia. There are few reasons behind doing this research work. One was the area, because South and East Asia is the one of the region which is home of more than around 50% Earth population. The second one is this is the Earth system model. UKCA is a Earth system model. I'm going to talk about this a little in later slides. And the third point is model evaluation is really important because the modeling work is really important for the future. Comparing model with the satellite is something apple and orange comparison. It's not easy. And if we have to do it, we have to deal with some errors. The first one is there are many errors, but I'm highlighting only 2 here. One is the model world is a regular world where the satellite overpass Earth depending on its swath. So it is irregular. The second point is the clouds are not well represented in the models. So clouds are different in the models, where in the real world it's are very different. The third point is the sensitivity of the satellite is different. So satellites use generally, partly I'm talking about NO2, and its sensitivity is different than the others one. So it is very much for in the middle range, it is very sensitive. It's not sensitive towards the surface. So to deal with all those things, we have to deal with the errors, and we have to apply averaging kernel, right time sampling. We have to filter out the cloud covers, all in the modeling work. So this is really crucial and important. A little things about the model. So the first half, you can see the upper portion, I'm giving the model details. I'm saying the last point UKC is not with the ERA interim data set. So ERA interim data set is the 3 hourly data. What I did, I nudged the model so that model is going and checking the real metrology every 6 hours and nudging itself to create the real metrology. So it is a nudged one. So that was really challenging in when you're running a global climate model. Then the UKCA, why UKCA is important? Because this is the component of atmospheric component of UK Earth system model, which is in the CMAP6 experiments. And NO2 is important because this is the oxidant. It is very important for the OH radical, methane, and ozone in the atmosphere. So if the NO2 is not right in the model, it is going to impact many other things. So this is important. That's why this whole idea was. So let's go to the what I did in the modeling work. So these are the this is the South and East Asia. I've already said this is the home of around 50% of the world population. And you can see the hottest spot of NO emissions. So this emissions is coming from the AirChem Collins 2017 is one of the most advanced on the reliable emissions which we have so far. And this is the difference from 2005 to 2015. So you can see there is an increase, and this was reported, well reported in the McKenzie et al. In 2017. So this is I am putting in the model, and this is happening in 2 big nations over this region, India and China. So you can see here one point, this is after 2011, the China's emissions was increasing. And after that, it started decreasing. Whereas in India, it is increasing continuously. So up to these 3 slides, these also are going in the models. Now let's see some results. And this is really interesting results. I'm saying 2 things here. One is the OMI is the instrument that is on board on the NASA's Aura satellite. And equator crossing time is 45, or so it is afternoon it crosses it. So in the modeling world, we have the chemical time step of this model is 1 hour. So I am outputting it 1 hour's voluminous data, terabytes of data. And it is every hour we have data. And that's why we have the diurnal profile. So here I'm just sampling, providing 4 seasons and how, when we are sampling it, where is the OMI overpass time? So OMI overpass time is, you can, if you'll see this data closely, you'll find particularly this point, the concentration is minimum in nature. At the time the satellite is overpassing the region. But, and this is the signal, this is the right hand side, you can see how it is vertically distributed. And this model is 85 kilometers. I'm just showing only up to troposphere. So summer season is different, winter time is different. And I'm going to highlight where the model is doing wrong. So you can, so the next plot, particularly this one, this is also the diurnal cycle. It has a few more information. It is a diurnal cycle with the concentration. With that, OMI overpass time is there with boundary layer height. So you can see when we are sampling it, the concentration is minimum. So let's see how model and observation are different and how much it is reliable. So the blue line is we are getting from the models. This is the UKC model, monthly mean data. Then I did sampling at the right time when the satellite is overpassing. So it is around afternoon, and we have seen there is a huge difference. The data is going down. But see, in the winter time, there is still a gap. And I have also said in the previous slides that averaging kernel is very important when you are, because this is a weight you are applying on the models for satellite data sensitivities. So average, when we apply averaging kernel, you can go it further close to the observation. So the green line is the observation. That is, and you can see there's a lot of issues with the light retrieval algorithm is there. So that there is some issues too. So let's see some results. What is happening in the South and East Asia, in the OMI, the real world with observation, you can say now, but what is happening in models? So in UKCA, particularly, I'm just showing only the monsoon, you can say, and the winter months, and you can see the model is overestimating it. So there is some issues with the model. Although the concentration is increasing in the winter time over the satellite data also. So this is the observation is in the left side and the model is in the right side. This is the difference band, and this is really important one, how it is things are changing. So you can see the your ammonia plot, you can say the IGP is the highlighted one. Here also IGP is highlighted because N is really high. Model is also overestimating over this region. Eastern China is also have some problem. These are the scatter plots, and in this one, two things I am highlighting. One is I am just plotting two lines. You can see the scatter plot two lines. One is the three lines, in fact, 1-to-1 line. Green line is the 90% data I'm plotting, and the red line is saying the overall data, all data. So 90% data means I'm just filtering out first 90% data, higher values I am not plotting. And you can see the differences. So here we can find what are the problem with the model. Come to this one. So as I said, the China and India story are different. And up to 2011, China's emission was increasing. There was an increasing trend and then was start decreasing. So I have plotted the trends. What is happening in the observation? What is happening in the model? So, okay, model is doing seasonality is okay. And this is the trend so for 2005 to 2011, and then come to the 2011 to 2015. So these are the trends. You can see there are decreasing trends in the model as well as in the OMI. This is, so there are something problem in the model. And this model is an all system model, which is going to used by the symmetric experiments, all 26 models are there. So what is the problem in the model? One reason, maybe you can say this is due to the boundary layer height. Here I'm highlighting 3 points. One, what is happening in the model and what is happening in the ERA interim data. Remember this model was merged with the ERA interim data sets. This is my last slide. So ERA interim data set and ERA 5 data has 1 hour resolution. And you can see here that after sunset, what is happening? There is a sharp decrease in the planetary boundary layer height in the model. So the all concentration is all to emitting in the model is coming in a shallow layer. And when we are sampling it at afternoon, it has that impact. So that might be one reason, but this is not the only one. Definitely, there are many other regions when we are doing the sampling. Satellite data sampling, there might be some issues with the model chemistry. Model is not, although this emissions we have used is, there is no diurnal cycle in this model. So model is doing this all calculation itself. So the diurnality is coming okay. So this is this summary. Model is overestimating, there are the errors, and thank you. Yeah.
So from which year did you observe declining trade
So this was reported in Mejeki et al. Also. So this was 2011. After 2011 to 2012, the China's emissions was NO emission, particularly I'm talking about, it starts decreasing afterwards. So there are some trends. We did some trend analysis for that too. So after 2012, that is reported in the literature. We're talking about India. So in India, up to 2015, it is increasing. But now if you say it is decreasing, so up to 2015, it was increasing. So it's a little delayed one. Yeah.
So, this, actually, the question which you answered, he has shown what is happening in Southeast Asia and what we are observing tomorrow.
Yeah, that is one part of
So, yes, so that is the main part.
the part.
Because depends on the precursor gases. The ammonia which is in gas phase depends on the precursor gases that is NOx, SO2. So basically, he has shown before.
Very good. I appreciate your observation. You are at least attentively.
Obviously, yeah.
Very nice.
That is how work and she was doing it. Okay, thanks. Anyone else? I am in the middle of lunch and so Soumya is there. Thanks a lot.
So, last but not the least, the speaker is Soumya Singh on exploring dynamics of atmospheric ammonia in India, distribution, trends, and source analysis. Soumya.
Hello everyone. Thank you for coming for my talk, and it's because it's a flash oral talk, I'm going to give you the helicopter view of the data set I have chosen to talk about. So as you can see the title here, I will just, okay. So this is the one slide I always talk about, and it's just showing you, I mean, we have talked about a lot that why ammonia is important, what are the impacts, but this, I really want to start with this, showing all the global ammonia hotspot. And you can see India, of course, in the red zone, and not only India, there are other regions as well. And the red actually showing, in the past, it's a satellite data. It's not my work, it's satellite data. You can see the sources. So it's because of the increased fertilizer application and other reasons, they have seen the increase in the ammonia concentration. And there are blue zones where they have seen the decrease of the ammonia emission. So why and how it all started? I mean, I'm now I have brought you back to the ammonia zone, and now I really want to emphasize that why this is important, why I'm going to talk about all of this. So first, ammonia is a pollutant, and it's important to talk about. And we have been, I think, many talks actually emphasize whether it's based on the modeling, whether it's based on the satellite data, measurement data is important. We need that for the validation of the model or even for the satellite data. Then in this reason, limited observations are there. But why that is the case? Because one is the measurement is challenging. As Alex already shown you the most, I mean, that's the most difficult measurement we have done using the QC till dust. And now, what are the data actually available? So far, I have been working on the measurement of the ammonia, but I really wanted to look at what are the data actually available and versus what are the quality data we have or actually we need. So let's look at what we have available. So this is basically, we scrapped with my colleague Sarah, we scraped this data from 2010 to 2022, I guess. Yes. So you can see this is the Central Pollution Control Board data set. These are dots are the monitoring stations, and that's also related with the concentration level. But what is striking and interesting that this much data is available. These are the sites. So until now, I think until now we have 300 plus monitoring sites in India. But so with that, what are we doing actually? Okay, so I thought that it would be really nice to look at the IGP and non-IGP trend, because we have been always, and we have Martin here sitting, and I mean, there are so many paper people have given reference that, the IGP always showing as a hotspot. But I was interested to look at the non-IGP region as well. What are the differences? So just to remind you that this is not the final, it's just a basic analysis. So what you see here orange is IGP, and the numbers you see, actually, these are the monitoring sites we have available. Okay. So then, the goal is to also look at the source variability. So I was very interested to see what are the small city versus an actually large city, and what the population can be even factor of that. And just to highlight that none of the sites actually are located in rural sites. And I mean, again, this is very basic analysis. We wanted to look at if we split the data and, see that we have two different kinds of seasons in India, weather season, classification, and crop season. So do we see any significant distinct features? So if you can see the IGP one, we definitely see the winter is high, the highest, winter shows the highest concentration. And I guess this is because in IGP there is a very distinct feature of the weather pattern. And in case of the crop season, I think I will skip because we haven't seen really any different level. Yes, it's the conclusion. And now this is the basically, you see this monitoring site increasing. It's actually the part of the National Clean Air Program for by the Government of India. And it's really important to understand what are the quality data. So just to highlight, just to give you a perspective, we have done, my supervisor is sitting here. We have done this impingement-based measurement in India. And then we have done in Canada the AD covariance with using the QC data. So where are these regulatory monitoring analyzers? I think I should move this here in terms of the cost, in terms of the quality of data. So I guess I wanted to really raise a question that if we are putting these many monitors, are we looking at the data carefully? And if yes, then what are the data actually telling us? That's all. Thank you.
Ask one person, you can discuss with me. Lunchtime, quickly. No one time, no question. Thank you.
I quickly request the co-chairs to please felicitate all these speakers, if you could.
Oh.
Thank you. Dr. Clear, please. All right, the first speaker, Dr. Dibakshi. Next, Dr. Lupali.
Yeah. Which side? Please come, please. Take photo. No many.
Dr. Ajitya Deshpande. Dr. Maya. Dr. Budeka Veeram. Dr. Alexander Mora. Dr. Kumar Shande, please.
Thank you very much.
And finally, Dr. Sonia Singh. I now request Dr. Suhail Khan from the organizing committee to please felicitate the co-chairs. This session now comes to an end. The time for the next session would be 2:20. We shall be 20 minutes. Thank you.
Sorry, I did from the organization. Because the machine, the coffee machine is off and we really need a coffee before starting. Is it possible? Thank you.
बोलना मैडम कितने आवाज चाहिए
I'm plate here na baहर हाँ सही प्रेजेंट करेंगे
इस अपलोड कर दिया आपने ऑलरेडी?
हाँ इसमें इसमें
या हमारी है ना ये तो पेन ड्राइव
हमारी है इसमें हमारी
माय व्हाट नेम
पीटर
यार एक सेकंड यार you can check it इसमें ही है
अभी इनकी है ना हाँ
खा लिया आपने खा लिया इसकी
क्या किया ना
थी।
presentations?
हाँ यार
I
अभी
hope
आई
कि-
थी ग्रोफमैन की आई थी और मार्क सटन की कितने डाली मैंने ये तो नहीं है
Certain का number क्या है? अच्छा ये last है।
सटन इज 320 एक बार मुझे वो दिखाइए
यहाँ पर TBD है तो मतलब ये होगी टॉप नंबर 6।
320 है ना
हाँ टॉप 6।
मार्क सटन नाम है
Yep.
ठीक है और एक ग्रोफमैन की फर्स्ट है इसकी भी आ गई मुझे लगा नहीं आएगा
सिर्फ 1 लगा दो उसके आगे इंस्टेड ऑफ।
क्या हाँ, वो। कैसा है भाई लंच यहाँ का अच्छा बना है
चिकन खाते हो?
छोड़ दिया
है।
तब से मेरी माँ बोल रही है सुन लो यार इनकी नहीं खाती मैं चिकन मैं जिम के लिए कर रहा था वैसे तो पसंद नहीं है ओके अब मैं स्क्रीन शेयर अभी ऑन कर रही हूँ so that I don't forget. और भाई एक बंदा है
हाँ वो
मार्क सटन कौन होता
एक पीछे गाना चला रहा था वो छोड़। हाँ एम प्लीज क्या मार्क्स है?
है पागल उसको भाई जब उसके भी रिप्रेजेंट होना याद रखे कुछ भी नहीं आना चाहिए आसपास वो पागल हो जाएगा कुछ भी हाइड नहीं करना चाहिए बढ़िया मैं स्क्रीन ही शेयर कर रही हूँ
या
मेरे को स्क्रीन शेयर बंद करके तब चलाया करो उसे।
रुको एक सेकंड
क्या करना है तुम्हें?
कुछ नहीं करना ये तो कहाँ गया आइकन वीडियो चल रही है माइक भी चल रहा है सब चल रहा है ना मेरा ठीक है
ऑडियो रिकॉर्डिंग करना है।
लैपटॉप से चलाए रखना है। हाँ ऐसे चलाई हो।
एक सेकंड मेरे को वीडियो क्यों नहीं दिख रही मेरी
क्या करना है?
रुको रुको
अरे दिस इज एयर पोल्यूशन, दिस इज नॉट नाइट्रोजन।
वो नाइट्रोजन 3 तुम्हारा वाला
नाइट्रोजन फ्री
तो एयर पोल्यूशन क्यों नहीं आ रहा
क्योंकि
आ
या
अब मैं आ रही थी इसको अगर मैं इसमें डालूं एक सेकंड ठीक है अभी इसी किया ये है ना हाँ पीटर बर्गमैन ठीक है कर लिया मैंने आपका लेट खत्म हुआ ना
कितना टाइम हो गया इंटर्न करते हुए विद दोस गाइस? कितने इवेंट्स करवा लिए?
पार्ट में ले ली फिर कॉलेज में तो काफी अरे अरे
अरे
चलो
Okay, or you have to, the next thing is I have to, yeah, because it just changed. It's not. So I just wrote it down. And how strict are we gonna be with the book to pull people on? I am soft, but I don't want to apply more pressure.
Sorry, this turn is on and then 2 minutes before, 3 minutes before, 23 minutes. No, I didn't. I will.
And do they, is there, what's that time?
Yes.
Okay, you.
First, we have to go, right?
हाँ ठीक है
We can sit. Yeah, so the flash talk will be a little different because one person has two slides. And people have, he's gonna have one and we can stop. Yeah, so Mark will be talking in the last two, but he's just gonna have one talk to say.
मैं ऑरेंज खा के आ रही हूँ मैं इसलिए बोल रही
And so how that's.
We didn't have ज़्यादा lunch for you
हूँ नहीं यार मेरे को स्वीट खाना होता है मैं हेल्दी खा देती हूँ बस
वही गुलाब जामुन नीचे में अच्छे हैं पता है अच्छे हैं मजा
भाई अच्छे ही होते हैं अच्छे हैं यार सब ठीक है थोड़ा हाइड करना ठीक है
Okay. What's your name? Isha. Okay. Ayush. Okay.
So we are here, so the course is small. We are 5 people, 6 and the business are.
You all will deal with people's thoughts and all of that. Yeah. Yeah. Between us, we will.
उधर देखना है
Oh,
so we like we have
probably at the end of the whole.
अभी
Because
आ गया
in my experience, everybody has been running a little bit off. And so.
ये जो करते हो ना दिखाता हूँ शेयर स्क्रीन किया उसको स्क्रीन 2 पे जाना है आपने ये सिलेक्ट किया है
पर एक बात तो हमेशा ही आएगी सेम
हाँ जैसे ही आप प्रोजेक्शन में डालोगे
ये है ना पहली बार पीपीटी में स्लाइड से डालना फिर
ये वाला है ना ये ठीक है मैं स्लाइड शो डाला ठीक है ये स्लाइड शो आ गया अब मैं क्या करूँगा स्क्रीन पे जाऊँगा इसको सिलेक्ट करना और स्क्रीन 2 पे जाऊँगा ठीक है एक बार उधर चेक करूँ ठीक है ना
एक सेकंड से वो मेरे Zoom पे ना जैसे हम इस पे गए पीपीटी ये वाली है ना वाला स्लाइड पे डाला ठीक है अभी टैब कैसे चेंज करते हैं देखो
slide.
नहीं
अच्छा, you want all
मैं गवा रही हूँ क्यों आ रही है
Want to check? Put it in the presentation mode. It's hiding from you.
getting used to it.
Okay, so if you hit the W key, it's a screen wipe.
Oh,
And then if you want to come back, you just hit any key, it comes back. Or if you want to make it black, you just hit the black key. Turn it black.
that's new.
And sometimes it's useful, otherwise you stop sharing.
Yeah.
So anyway, if you want to leave it black or white, you can.
Thank you so much.
Thank you.
We have a time or
We can start right away.
Oh, okay.
So
I know everything's running a little late. So
We'll just wait for 2 more minutes for people to come.
so why don't we just say we'll start right at
2:30.
अरे यार, मैंने सीखा था रुको
जरूरत है इसकी
क्योंकि मेरे Zoom पे पूरा स्क्रीन आता है फिर सर ने बोला है मेरे को क्या भाई ये कुछ तो होते हैं ये होता है ये होता है ये होता है ओह थैंक्स गुड गुड वेट अब शेयर स्क्रीन करी अब हमें स्क्रीन 2 शेयर करनी है ये तुरंत दबा के नीचे वाले है ना आइकन्स तो पहले पीपीटी पावर स्लाइड शो मोड पर डाल दो फिर नीचे आइकन्स लेके आओ रूम पे जाओ ये स्क्रीन के साथ वाली ये वाली जो पूरी पूरी इस पे क्लिक कर अरे इतना तो कर लेता है तुम पागल है मुझे
अरे अभी भी यही आ रहा है
नहीं बैठ आ गई
Start? Yeah. Okay. Welcome everybody to Third Pollution 3. I'm David Cooper from Western Washington University, US. I'm one of your co-moderators. And welcome to this conference. And so we will, just for the logistics for the speakers, our first speaker will have 20 minutes total, 15 minutes for talk, 5 minutes for discussion. And the speakers after that will have 15 minutes total, with 10 to talk, 5 for discussion. The people will go off when you have 5 minutes in your total time. So just be aware of that. Try to allow room for questions. So without further ado, because I know we're running a little late, our first talk is by Peter Brockman, biotic regulation of nitrogen gas emissions in temperate agriculture.
So I'm going to talk about nitrogen gas emissions, and I'm going to highlight some data from a couple of former graduate students. I'll try and point out their contributions as I go along. Now, most of the nitrogen gas talks at this meeting this week have focused on nitrous oxide. And I'm going to ask you to think about dinitrogen for a little while, as well as nitrous oxide. And so in this talk, I'm going to first start by talking about the differences between N2 and N2O flux. They're regulated differently. Their controls, the methods, their importance is a little different. I'll then talk about some data on spatial patterns of nitrogen gas emissions with the focus on hotspots or particular moments, particular places that have a high flux, and also talk about
temporal patterns and a little bit about hot moments or small bits of time that have very high flux. So there are two processes here that account for our nitrogen gases. So we have denitrification, which is the anaerobic respiration of nitrate and nitrite and produces three nitrogen gases. So here, nitrous oxide and dinitrogen. These are mostly heterotrophic organisms. And then the other big gas process is nitrification, which is an aerobic process, very strongly driven by ammonia, the concentration of ammonia. So this is the oxidation of ammonia to nitrate, and nitrous oxide comes off as a byproduct in that process. And because nitrification is very strongly driven by ammonia concentrations, it's also very strongly driven by fertilizer inputs. And so lots of our nitrous oxide studies focus on fertilizer inputs as a driver of nitrous oxide emission. But these are very, very different processes. So denitrification produces a lot of nitrous oxide, but it's an anaerobic process. It's a heterotrophic process. It has much different controls than nitrification, which is a chemoautotrophic process and an aerobic process. So we need to remember that these controls are different. We also have to remember that the N2O flux is very small relative to denitrification. I'll present some data on this. And so we're interested in N2O flux because it's a greenhouse gas, but we have to recognize that it's a relatively small flux in the nitrogen cycle. The spatial and temporal controls over denitrification and nitrous oxide flux are very different. And the N2 flux is important because it tells us a lot about where and when we might expect to get nitrous oxide flux, but it's also important in its own right. So as we talk about managing N waste, this production of N2 is considered to be a big component of the N waste. And if we're interested in quantifying and reducing N waste, we need to understand N2 flux to denitrification. Now, there's two reasons why we know more about N2O flux than denitrification. The first is we're very much interested in greenhouse gas fluxes, and nitrous oxide is a greenhouse gas. The other reason is denitrification is very hard to measure, much harder than measuring N2O flux. We're trying to measure a small flux of gas out of the soil into the atmosphere that's 80% N2. So if you have any experience with laboratory chemistry, it's the world's greatest contamination problem. Any small leak and you're not measuring anything. So many of our methods for measuring N2 flux involve alteration of the soil or physically unrealistic conditions. And so we oftentimes worry that our estimates of N2 flux are an artifact of the method that we've used. We also have very high variation in time and space. This is true for nitrous oxide, but it's more so true for N2 flux to denitrification because it's controlled by oxygen and by nitrate and by carbon. On all of those controlling factors have a variance structure, and you combine those together and you get very, very high variation. And that's led to this focus on hot spots and hot moments. If we can identify particular places or particular times where the flux is high, we can focus on those in our experimental designs and our measurement to improve our estimates of the flux. And then the other challenge with measuring N2 flux is scaling. So, and this is true for all of our microbial processes. We're measuring microbial processes, and yet we're interested in information at much larger scales. And so we have real problems in how do we go from, we have measurements that are highly variable and they're problematic measurements, and then we want to extrapolate them to large areas. So this is a challenge. So we don't have a lot of data on denitrification because it's hard to measure. But it's a very important flux. There's a tremendous amount, whenever we try to do nitrogen balances, we end up with a tremendous amount of missing nitrogen. This is one of my favorite examples of this. Is from an old paper from the 90s, but it's a watershed in Iowa. And over 22 years, they did an accounting of all the nitrogen in the system. So over 22 years, they applied quite a bit of fertilizer, and about half that fertilizer was exported in grain, easy to measure. About 20% was exported in water, either in surface runoff or in erosion or in groundwater leaching. But 30% of the nitrogen was missing. And they just say that it's lost or stored. Now, in our definition of nitrogen waste, this is a component of nitrogen waste. Just basically, we're saying that 50% is nitrogen waste. But we need to know, is this really being denitrified in this lost or stored, or is it going to some other pathway? So the need is very important for these N2 fluxes. And again, it's a big number, much bigger than the nitrous oxide, which is maybe 1% or less than 1% of the flux. So we have been using some relatively new methods to measure N2 flux. And in our system, we take soil cores and we put them in a controlled incubation environment, and we replace the nitrogen-oxygen atmosphere that we all enjoy. We replace it with a helium-oxygen atmosphere. And so once we have a helium-oxygen atmosphere, we can directly measure the flux of nitrous oxide and N2. And then these samples of the headspace in these cores go directly into some gas chromatographs where we can measure those gases. So this system allows us to directly measure N2 and N2O fluxes from intact cores without a lot of physical disruption or chemical disruption of the sample. And you can see here's what these samples look like. There's a soil core in a jar, and the samples are automatically introduced into a gas chromatograph to make measurements of these flux rates. We've also gotten better at measuring hotspots and hot moments in the field. So we've been using, and many people are using these oxygen probes so we can measure oxygen levels in the soil. So here I've got a site along a stream. I have 4 sites along the stream. The red sites are farther away from the stream. The soils are drier and they have higher oxygen concentrations. These are soils near the stream, wetter, much lower oxygen. And with these oxygen electrodes, we can get high resolution in space and time measurements of when denitrification and N2 flux are likely to be high. And so the new soil core methods and these new sensor probes are helping us in our studies. So let's show some data. I'll present some data on spatial patterns and then on temporal patterns. So one of the great controllers of variation in space is topography, landscape position. And many of us have seen landscape like this where there's a small stream at the bottom of the hill. The soils next to the stream are much wetter, and we expect that the lower positions on the hill will have higher denitrification flux than the places higher up on the hill. And this is work that was done at a long-term agricultural research site run by the United States Department of Agriculture in Pennsylvania. And so here we have a couple of years of data on N2 flux and then N2O fluxes below. And you can see the first thing you notice is that the N2 fluxes are much higher than the N2O fluxes. So the axis here goes from 0 to 3,500 for N2 flux, and the N2O flux axis only goes up to 150. So there's really a lot of N2 flux. The other thing we see is that, that's the wrong graph. The other thing we see is that the low positions have the low topography, the wet spots have higher N2 flux than the higher spots as we expected here. We don't see that topographic pattern with N2O. So we see the controlling factors of denitrification are different than N2O. And then we also see that there's a variation in N2O yield, which is what we're very interested in from a nitrous oxide point of view. And the low spots, the wet spots, have much lower N2O yields than the spots higher up on the slope. So here we're learning where is the nitrogen flux in the landscape the highest and where are the N2O yields the lowest, and that's useful information for our nitrogen waste goals. We also have variation in crop type. So in the same watershed, we have wet riparian zones dominated by, these are nitrogen absorbing crops, riparian crops. So we have a conservation reserve program grass mix. We have miscanthus, we have switchgrass. These are grasses that people are planting to absorb nitrogen. And we're interested in how do they stimulate nitrogen flux, either N2 or N2O. And we can see that there's some real differences, that the Conservation Reserve Program mix and miscanthus have higher denitrification rates than the switchgrass plots. We also see they also seem to have higher, and there's also quite a bit of variation in N2O yield amongst these sites. And in particular, we can see that this miscanthus, which is a very impressive plant, it grows very tall, very fast, it absorbs a lot of nitrogen and has very low N2O yields, which is useful for us. Another source of spatial variation that we've been going after is confining layers in the soil profile. So many of our soil profiles have hard pan layers, either caused by tillage or by natural factors. And as water moves down through these soil profiles, it hits the confining layer and it hangs up and creates a wet area deep in the soil profile, which you have long suspected might be a hotspot for nitrogen gas flux. And so these are data from soils in Missouri, another one of these long-term agricultural research sites. And we take cores from deep, these are from about 40 centimeters down in the soil at the confining layer, so very low organic carbon levels. And this is N2 flux and N2O flux. Again, the N2 fluxes are in the thousands The N2O fluxes are less than 100. And surprisingly, these deep cores have higher denitrification rates, higher N2 flux rates than the surface cores. The surface cores are the red, and these deeper cores are the blue. And indeed, this reveals that there is a hotspot of N2 flux deep in these soil profiles where water is hanging up. And also that's interesting is that these confining layers are a hotspot for N2 flux, but not for N2O flux. So we look at N2O flux, you see it high in the surface soil, but there's almost no detectable N flux down in these confining layers. So here we have in the soil profile a hotspot of denitrification and N2 flux, but with almost no nitrous oxide associated with that. I'll also talk a little bit about spatial patterns, some hot moments, and then some seasonal patterns. So hot moments are the idea, and are the idea that something in the soil environment changes and it creates a flux or a hot moment of denitrification. And many people have studied drying and re-wetting events. So we were at another one of these long-term agricultural research sites, this time in Georgia in the USA, and the soils were dry. And again, these are soils that have a hard pan layer at about 50 centimeters. And so we're sampling before when the soil is dry, and again, these hard pan layers, these layers deep in the soil profile, have higher denitrification N2 flux than the surface cores. And then we wet these cores to simulate a rainfall event, and we got a hot moment or a burst of activity. We see that both for N2 as well as for N2O. But again, as in these Missouri studies, the confining layer is a hot spot for gas flux, and the drying and re-wetting event is a hot moment for gas flux, but not for N2O. So the N2O flux of these confining layers, they're producing a lot of N2, but even after re-wetting, they didn't produce any N2. So again, we have a case here where we have a high nitrogen gas flux, and it's a very clean flux, and that the N2O yield is very low. And the other example I wanted to talk about, and this is the work that was led by former graduate student Maya Almaraz, which was looking at seasonal patterns and controls of these gas fluxes. So these were two sites, a site with sandy soils in Delaware in the United States and a site with more fine texture soils in Pennsylvania in the United States. And Maya was very interested in seasonal patterns. So if you look at just after fertilization in the middle of the season, and then you look at after harvest. And for those of us interested in nitrous oxide, this is when we make our measurements. We're very interested in fertilizer-induced emissions, and most of our nitrous oxide measurements occur at this time, less in mid-season, and almost no one makes measurements in the post-harvest season, except for people who are making year-round measurements. And the hypothesis was that the seasonal patterns of N2 flux were going to be very different than the seasonal patterns of N2O flux. The N2O flux was going to be driven by the fertilizer emission, and the N2 flux might be driven by changes in water and carbon availability as you got into the later season. And that's certainly what we saw. So this is N2 flux, and here's N2O flux. And as we expect, the N2O fluxes are very high after fertilization, and they're very low in the middle of the season when the plant is growing, and they perk up a little bit after harvest. The N2 fluxes, of course, are much, much, much higher than the N2O fluxes, and they are highest in this post-harvest season in the fall after the plants have started growing. And so the idea that the difference in these seasonal patterns, the N2O is driven by fertilizer input, and the N2 is driven by plant senescence and carbon and water flux. And just you look at this picture and you think about our crop fields after they senesce, the plants aren't growing, so they're not competing for water, they're not competing for nitrogen, they're producing a lot of fresh carbon. And so it makes sense that an anaerobic heterotrophic process like denitrification is very high in the fall, whereas the N2O is at a different time. Okay, so I'm going to leave you with the conclusion that we need to think more about N2 flux and denitrification. And we need to do that because it's an important flux. And if we're trying to manage nitrogen waste, we need to understand these large fluxes. I think it's also going to help us think about nitrous oxide. And as it's a big controller of nitrous oxide, and where is nitrous oxide flux likely to be high and in space. So it's an important flux. It helps us think about nitrous oxide, but we have to remember that the controls are different, and we have to consider that as we design our studies going forward. Thank you.
Good time for questions.
I have a question. So do you think it's fair to generalize that when you have high denitrification to N2, your N2O yield is going to be low? Because the conditions, when you've got high denitrification, you've got everything you need to take things all the way to N2.
Right, so that would be the driver, is if you have really anaerobic conditions, you get complete denitrification and a tremendous demand for N2O as an electron acceptor. So in many cases, sites will absorb N2O from the atmosphere. So yes, if you have very high N2 flux and very complete denitrification, you'll get very low N2O yield. But on the other hand, if you have a high denitrification flux with some N2O yield, your N2O flux could be high. But I think yes and no. The answer. Way in the back.
How might the growers use this knowledge in practice? Like how might your farmers in the United States use this information to shape the emission criteria?
Well, so there are two things we're asking farmers to do in the United States. We're asking them to reduce their nitrous oxide flux. And as far as we're concerned, I think we have about 3 methods to reduce the nitrous oxide flux. You can add less fertilizer, you can add less fertilizer, or you can add less fertilizer. But if we're trying to halve nitrogen waste, that's a much more complex question, and that involves consideration of this denitrification. And so if the denitrification counts as N waste, we then need to spend some time thinking about how do we tighten up the nitrogen cycle as a whole so that we use more in the plants and we have less available for this gaseous loss or for this loss below. So it should help our farmers to think about how do they design their systems to be more efficient in nitrogen use, which is a more subtle approach than just using less fertilizer. We can tell people to use less fertilizer, but they say, But if we think about redesigning our systems to manage the whole nitrogen flow, it might help us to redesign the system and achieve multiple goals.
So I'm going to ask a supplementary question here. So looking at rolling topography you showed, so how might a farmer change his practice on that topography?
Well, so those sites that I showed you shouldn't be farmed at all because they're way too steep. But many of our farmers insist on farming on slopes. But it certainly allows you to identify areas of vulnerability in the landscape. Where might the fluxes be high? So in that landscape, the very bottom slopes where you're getting a lot of erosion and you're getting a lot of nitrous oxide potentially, they're converting those to these bioenergy crops, which do not get fertilizer. And so the topography, thinking about topography, also helps us to design better cropping systems and then to better landscape systems. Think, well, where are we really vulnerable to environmental damage, and maybe we should do different things in different places. But topography, and farmers like to farmers know more about topography than we do. They know what the water's doing, they know what the fertilizer's doing, they know what their plants are doing, and so that interaction with farmers, I think, can help us to understand and manage that.
Time for one more quick question.
It should be quick. That you showed that missing nitrogen at the start, 30%. Just wondering if were you able to find that mass balance? Yeah, able to confidently say that where that nitrogen's going.
Well, so that mass balance it's an old one, and it's one of my favorites because it was done it was very detailed. It was done by hydrologists. And so they had a very good accounting for all the nitrogen that went in fertilizer and all the grain that went out. They very carefully measured the amount of hydrologic loss. And so then 30% was unaccounted for. And so it could be accumulating in the soil, but this is continuous corn in Iowa in the United States. And we know pretty much that nitrogen is not accumulating in those soils. If anything, they're losing nitrogen. And then people have been measuring denitrification in Iowa since the 1950s. And they've never been able to measure such high rates that would account for 30%. So that nitrogen balance is a mystery. And I love to present it to students to go out and figure out where's the nitrogen going. Because and maybe the denitrification rates are high enough to account for it, which is why we measure it. But the mass balance, to me, puts a constraint on the limits of what we know about the nitrogen cycle. And again, if we're trying to reduce reduce N waste by half, we need to figure out where that 30% is. And so I really like showing that because it's very carefully done and it's long term. It's a 22-year balance.
Thanks, Peter. And our next talk is Alejandro Montero, talking about nitrogen deposition over Portugal, the Fonda project.
Okay, yeah, that's fine. Okay. Perfect. Okay, hello, everyone. Hi. Okay, so now it's ready. So hello, everyone. I'm from Portugal, and it's my pleasure to present here and to share with you some this project that we have now ongoing, a European project that is related to the nitrogen deposition modeling over Portugal and Europe also. When I started to prepare this presentation, I was very glad because I realized that this work fits completely into this conference. That's my first time here. When I realized that at least 5 of these sustainable development objectives fits into our work. So this means that it makes sense to present it and to share with you here. So the origin of this project starts because there is a problem regarding nitrogen deposition in Portugal, in Europe, but if you look at Portugal, it's a specific area where the critical load, it's a very quite sensitive area where the critical loads are very easily exceeded. So we have anthropogenic activities as everywhere. We have ammonia and nitrogen oxide emissions, but we don't know exactly, specifically for Portugal, we have a lack of knowledge regarding the deposition. We know that we have emissions high because they're now at the moment they are exceeding our goals for the European emission ceilings. So we know that we have this problem, but in terms of the position, it's still very unknown. And also the future show us that this problem will probably increase because for example, shipping fuel will probably pass through the ammonia as a renewable fuel. So this will increase the problem. And so that's why this project, this twinning project funded by the European Union appears. And the main objectives are to make this knowledge in terms of the position modeling for Portugal, for our specific, and for that we build a consortium with the TNO from Holland and the University of Berlin from Germany, because they have developed already a quite complex modeling system that includes a deposition model with very detailed. So the purpose is to adjust and develop this deposition model for this specific area, Mediterranean area, that is very different from the north of Europe that they developed for, and also build some consortium that could be useful for future collaborations and international purpose. So the consortium is basically this. We are just quite young and motivated persons, and the project, the Fonda project, has a very strong purpose of the capacity building and training, but has also research and development goals. That's what I told you about developing this deposition model for Portugal. And this is the work package too, and that's why I'm here in the conference, not because of the training and the capacity building. So basically, this research will be based on the Lotto Zeros modeling system that was developed by the TNO from Poland. And this CTM, it's an open source 3D regional Eulerian CTM, but it has a specific development for describe the bidirectional surface atmosphere exchange of ammonia
and the so-called co-deposition effects between SO2 and ammonia. And that's why we choose these partners and the specific modeling system. So basically, for the ones that are familiar with CTM, it's easily to identify the inputs, the traditional inputs, the emissions, and then the Lotto Zeros model that also includes the process, the natural emissions, transport, mixing, chemistry, wet and dry deposition. We did the first applications of this Lotto Zeros modeling system as it is now, okay, with the deposition model included, but developed for specific areas in the north of Europe. But we apply directly for Portugal just to see what kind of values are we talking about for this region where we don't know, we don't have knowledge about these deposition values. And we don't know yet also because we don't have measurements. They are very, very scarce. And when we try to validate the models, it was impossible because it was one value in 1 year. We don't know if it's correspond to a daily, to an average day of the month. So nobody can tell us that. So we are still struggling, and definitely we need to include a campaign, a monitoring campaign in this project. But so we did this first application, we ran for Europe, but then we focus on Portugal with the nesting approach. We use the ECMWF as the methodological forcing, and we ran 2015 because it was the year that we thought that we could have some deposition data, which at the end we are not sure if we can compare it. But so we start to validate. Deposition are of course a consequence, a balance of concentration of what goes to the air and goes to what is deposited. So we start to validate also the concentrations. And what we found for the main critical pollutants, NO2, PM, ozone, these are the main critical pollutants for our region. Okay, of course, we have ozone problems because we are a Mediterranean country. And so we compare, and the agreement was was quite strong. Okay, in average we had some bias, maximum bias of 5 micrograms per cubic meter for NO2 and PM, and maximum 10 for ozone. So it was not bad at all. We have some in Lisbon and Porto, where the main emission sources are located. We have some less skill of the model that we have to develop, but we are sure that is related to emission data. Okay, here is the ozone that I want to show you, and here it's PM. Okay, PM it's where we have a really overestimation of emission. We have to refine our emission inventory, and this modeling application just confirms that. We already knew that, in fact, but this confirms that we need to work on this urban emission data. Okay, this is PM 2.5, where very much lower monitoring sites exist, but even like that, there was possible to see quite good agreement. And then we look to the deposition. Okay, here is the position over Europe, part of Europe, not whole. And if you see, and if you ask me why the north of Africa is included in this domain, because we have a strong influence of dust episodes, so we have to include it, otherwise it would be a real mistake. And then we look to Portugal, and if we look to the European, we don't recognize, we don't see strong values for the nitrogen deposition of Portugal. But remember that I show you this picture in the beginning of the presentation, means the critical loads, the estimation of the critical loads. And so this is really, and you know that this is really important. Even we don't have so high deposition values as, for example, Belgium and Holland are one of the hotspots in Europe are these countries. We are in a very sensitive region. So these deposition values are probably too high. And now I want to talk a little bit. I'm really sorry not to present a comparison between the observations and the position modeling, but it was not possible to get monitoring data, which is really very sad. But so I want to talk about the developments that we are now doing. So basically, two types of developments we want to reach. One is the improvement of the deposition model for this specific area south of Europe. And we also want to solve the problem of monitoring data, how to solve it without monitoring, for example. So for the first goal, we are developing, thank you. We are developing a dry deposition model that includes 9 specific land use classes. And for that, we have a tier 3 approach that will allow us to have much more detail on the parameters to parameterize the deposition. How do we do this? Until now, this was only based on land use and vegetation map. But now we divided this into into climate zones. Okay, this will allow us to specify much better the vegetation map, and so we will have much more classes, and we will allow to increase the number of parameters to parameterize the dry deposition model. And if you want to know a little bit more about that, we can discuss further. About the other goal, we want to use satellite data. Since we don't have constant monitoring data constantly, continuously in Portugal, so we will use monitoring data. And these are just examples. Tropamine for NO2, Yazi for ammonia, and Sentinel-2 for props. So we will use this to compare with our model. So I hope next presentation that I will do for this project, I will have already the comparison with at least with the satellite data. That's my goal. I would like to take this opportunity just before finishing to tell you that we will have a spring school next month in our university, University of Aveiro in Portugal. And this spring school is completely related to this, to the subject that we are doing this. And I'm telling you this because there are still registrations open until the weekend. Okay, I know that it's quite soon, but if you have any chance to look at it, if you are interested, or if you have students, this is more for PhD, young researchers, of course. Here, just a brief overlook, and you can check on the web all the information, okay? So thank you so much. These photos are just to convince you to go to the spring school.
We've time for a couple questions. So rising deposition is declining rather rapidly in Europe. Is it true in Portugal? And does that make it much harder to model deposition when it's changing somehow?
We don't know. That's the question, because we don't have monitoring data that allows us to understand what happens in the past and what is happening now. We don't know. I don't know how to answer this, because we don't know. I think it was not mandatory, you know, it was not. So Portugal only do what is mandatory by the European Union, and it was not mandatory to monitor this, at least continuously, and we didn't. Thank you.
I'd like to know,
but I guess I can hear you. Yeah.
Yes, the deposition you showed about N total N. So the model which you are using,
yeah,
how much different types of nitrogen species you are calculating?
Yeah, it's a good question, because I didn't do the project. I'm the coordinator of the project, and I don't know exactly. There are several, at least 4 or 5, but I don't know exactly how many. But I can just check and tell you. Okay, sorry, it was PhD students that did this simulation, and I know the main species, but I don't know all. but I will check. Yeah, yeah.
One more question.
Thank you.
Okay, thanks very much. Our next talk is Hannah Getz, talking about mitigation effects of urea incorporation techniques on ammonia and N2O emissions depend on soil conditions.
Perfect. Yeah, thanks for the introduction. I'm Hannah Götze from the Thun Institute for Climate Smart Agriculture. And yeah, the work we've done is part of the NH3min research project. And I want to show you results of our studies in the lab on mitigation effects of incorporation techniques on ammonia emissions. So just really quick, a little background because we already heard about that a lot. Agriculture is the main source for ammonia emissions, and these emissions can form fine particulate matter that can be transported over long distance and then leads to eutrophication and acidification. That is really bad for nitrogen poor ecosystems, can cause changes of biodiversity and loss of this diversity, and is hazard to human health. And on the other side, the goal is to increase the nitrogen efficiency to protect our resources, and also really important to the farmers to save costs on fertilizer. Just little numbers. For Europe, I'm focusing on mineral fertilizer in this presentation. So in Europe, 20% stems from mineral fertilizer, mainly urea-based, and for Germany, it's 12%. And the goal for Germany is to reduce the ammonia emissions by about 30% by the year 2030 in comparison to the levels of 2005. So how can we reach this huge goal? There are different opportunities, maybe changing the rate or split application due to the crops need the right time due to weather conditions, but I will focus on the right place. So we will have a look on different incorporation techniques to reduce ammonia emissions and increase the nitrogen efficiency. Yeah, so incorporation generally is considered as an appropriate mitigation measure, but still the potential in reduction ranges a lot, about 50 to 80%, and the data is really limited on different techniques that could be used, different soil textures, or other soil properties. So we wanted to focus in this study on different techniques, different soil textures, and different moisture levels. And we didn't just have an eye on the ammonia emissions, we also wanted to have a look on N2O emissions to be aware on a pollution swapping. So yeah, the setup in the lab, we did four application techniques. The first was standard broadcast spreading, and then the incorporation techniques: surface, really loose incorporation, 2 centimeters deep incorporation, and closed slit injection. You can see it in the picture a little better. We applied the fertilizer into a slit and then rolled over by a wheel, and we had a look on 3 different soil textures. As I mentioned, they were really different, but soils that we also used in our research project: a sandy soil, a more loamy soil, and clay soil in 2 moisture levels each. So how did we measure the emissions? At first, ammonia emissions, we had open dynamic chambers with a high flow rate. They were continuously measured for 2 weeks. We used acid bottles to trap ammonia with sulfuric acid in it, and measured then the samples with an ammonia selective electrode. And the N2O was measured in another setup with a gas chromatography for about 8 weeks. So at first, the results of the ammonia emissions. Here is an example. You can see the sandy soil in the 2 moisture levels. Blue is the standard surface broadcast spreading, and in green, the different incorporation techniques. And what we can clearly see with from the data is that slit injection and deep incorporation had a difference in the emissions, that it mitigate the emissions, but the surface incorporation in dark green had no significant difference. So little deeper look into, before I wanted to highlight something, that the higher soil moisture increased the initial rate at the beginning, but didn't change the total loss. We have just this lag phase in the beginning when the moisture content is less. So yeah, as I said, little deeper view into the data. Here we see the different incorporation techniques, and it's the percentage of mitigation compared to the standard broadcast spreading. 3 different colors for 3 different soil types. Clay is blue, loamy green, sandy red. And the brighter color is always the lower soil moisture. And what I want to highlight here is that surface incorporation doesn't lead to a mitigation effect on all soil types. And the highest mitigation was reached with a closed slit injection on the loamy soil. And in generally, we can say that, for the clay soil, all the different techniques you can see in blue lead to mitigation, a mitigation effect. The reduction for slit injection differs between 43 and 78%, and for deep incorporation, it varies a little more from 70 to 82%. Just really quick, that we can see that it's really significant, that it has a significant effect. I'll turn the pointer on. So for the sandy soil, both moisture types, the surface incorporation had no significant difference. When we look at the clay soil, we see all the incorporation techniques had a significant difference. That I wanted to highlight just again. And let's switch to the N2O data we got. Here we can see all 3 soil types. We can see that from the clay soil, we had just background signal, no N2O emissions. And from the loamy soil, we had less N2O emissions than from the sandy soil. But what is really interesting here, and what I wanted to highlight, is that in green is the lower soil moisture content, and in blue, the higher soil moisture. So higher soil moisture lead to higher N2O emissions. And what is really interesting, because I'm talking about incorporation, the lighter color blue is the surface incorporation. So we can see, still the pointer on, here is a difference between the standard application and the loose incorporation. So here we have an effect by that. So surface incorporation can stimulate N2O emissions at a higher soil moisture level on these 2 soil types. And to conclude, the incorporation techniques were effective as a ammonia mitigation measure. On clay soil, all incorporation techniques were really effective for sandy and soil, and when the conditions of low soil moisture are present, we should definitely consider maybe also a deeper incorporation or slit injection. But in the end, we should be aware of the N2O emissions. They may limit the use of urea incorporation to reduce ammonia emissions, and maybe then we should, it should be, there's a require of other mitigation measures. So I think I have a little time left to give a little outlook. We also compared with inhibitors. So again, this slide, there's also the right source we could choose. So using a urease inhibitor or nitrification inhibitor is also an option to reduce the ammonia emissions and increase the N efficiency. Perfect. And just a really quick view into that. It's also a large trial, but I want to highlight just that we can see here the urea standard application, here the different mitigation options, the slit injection and the inhibitors. We can see every inhibitor leads to a mitigation, and it was a significant effect. In numbers, it was about 45% for the inhibitors and about 75% for the slit injection. So there, the slit injection performed even better. And Jim, yeah. So all the mitigation options mitigate the emissions, but the lowest was reached by slit injection. And yeah, we will see here, I have to be fair with the nitrification inhibitor, there's a different kinetic, and this trial was really long to see really what happens over the time. So there is still a gap between the two inhibitor users. And so just to say that. And yes, thanks for listen to my talk. I'm happy to take questions. I have also N2O data for the last trial. We could talk about that maybe in the break. And as I said, this work is part of the NH3Min project, and you can scan the code. That's our website. And if you want to hear something about measuring methods, you can see here alpha sampler or passive sampler in the field. Please come to my poster. It's poster number 16. And I want to thank all the help in the lab, Melanie, Carsten, and Leah. Big support. Thank you.
Good time for a couple questions. Thanks for today's presentation.
Thank you.
My presentation is just for clarification.
Yes.
On the measurements. For ammonia measurements, I think you mentioned that for measured continuously. For 2 weeks.
Yeah, right. Oh, that wasn't too far.
You meant the vacuum chamber was kept paused for 2 weeks. So of course that would that might affect the exposure, might affect the ammonia. You meant you want to expose it when it's something and then you open it.
Yeah, really good question. I could talk a long time about the setting up of this trial. The little jars are prepared with the soil, then fertilized, then closed, and they are connected to air running through it all the time. And we have after the jar, we have another bottle with the sulfuric acid in it. There, the air is pumped through, and so all the air got captured, and I closed it really quick when I want to exchange samples, and then directly have a new prepared sampling bottle, so I can take it to the lab and then do the measuring with the electrode. Thank you for the question.
Sorry, our microphone is not working. Thank you for your nice presentation. I have two questions. The first one is, have you compared the total nitrogen losses What are like the losses that you calculated?
Yes, yes.
Different one.
I want to add something. We have also, we did also soil samples to try to close all that. And yeah, but it's too much data for short presentation.
And then my second question is, you excluded the plants.
Yeah.
Then what's going the growth?
Yeah. Yeah, perfect question. It's a lab trial. Yeah, we should definitely also have a look into that in the field for sure. That's a really good point.
We're actually out of time for questions, but I encourage you to talk more with Matt afterwards. Thank you very much.
Okay. Announcements, the end of the session. That we replace the So our next speaker is Andreas Kaczowski on information.
Because I heard you, but I didn't quite hear the message. That is correct. At the end of the session, I will also stay and enjoy the time.
Okay. You use the other tools to know. Okay. Okay, so hello to everyone. I'm quite delighted that I have the honor to present some thoughts, some ongoing work which I have together with colleagues from the German Environment Agency on the knowns and unknowns of the use of nitrogen transformation inhibitors. And I, of course, want to acknowledge my co-authors here in this context. So we heard already a little bit about this product by Hannah in the previous talk. And I just want to dive into the content. So what do we know about these compounds? Of course, nitrogen transformation inhibitors, it's just a term I like better than nitrogen inhibitors, because what they do is they inhibit the transformation. And when we apply fertilizer, very often the emission losses or all the nitrogen losses, they stem from after the transformation of the product that we have applied. So mainly it's about two processes. That's one is about ammonia emissions. As you know, when you apply ammonium or urea to soil, you can create ammonia emissions, and that's mainly due to the original soil pH or due to the increased soil pH by urea hydrolysis, catalyzed by urease. Secondly, when you apply ammonium to the soil, you can create N2024 emissions due to its transformation to nitrate by nitrification, or subsequent transformation to nitrate. Both processes, nitrification and denitrification, can cause N2024 emissions. And certainly, there are also another inhibitors existing, so-called denitrification inhibitors. They're also existing on the market, but I don't want to treat it here because it's just upcoming products. So with respect to the first process, how do urease inhibitors work? They slow down the urea hydrolysis process, and thereby also have a strong effect on the pH increase in soil and thereby reduce ammonia emissions. Secondly, by nitrification inhibitors, they can be both synthetic and biological. Here I mainly elaborate on synthetic compounds. Short comments about biologicals. And they mainly inhibit the microbial enzyme MOA and thereby reduce, which is responsible for the very important role in the nitrification process, and thereby we reduce N2024 emissions and also subsequent nitrate leaching. In this table, I summarize what kind of expected effects exist for inhibitors, just expressed in effects on greenhouse gas emissions. And yeah, as I mentioned, for the NIs, for the nitrification inhibitors, which also exist in double inhibited products, and you have urea with urease and nitrification inhibitor, we expect, of course, that we have a reduction of N2024 emissions, and similar things are discussed for urease inhibitor, a little bit more controversially. Methane, we don't know much about it.
And for CO2, you can expect that there are some trials where you can see that microbial activity might be reduced, and then you have an overall reduction of CO2 emissions. The other effects I talked about, I address here as indirect greenhouse gas emissions. So for the urease inhibitor and also the double inhibitor, we expect, because the double inhibitor also includes urease inhibitor, reduced ammonia emissions. And there's a lot of evidence, and we also show some data that we can have increased ammonia emissions when we just have nitrification inhibitor with ammonium or urea fertilizers. And there are some studies showing that we can have reduced nitrate leaching, and also because of lower fertilizer losses, we can have, by all these compounds, higher yields or higher nitrogen use efficiency, which result in lower fertilizer requirements. Just to motivate a little bit, yeah, what is at stake? So in Germany, just an example, about more than 90% of ammonia emissions stem from agriculture, and 10% of those from mineral fertilizers. Hannah told some data, 12%, predominantly by urea. We have very ambitious goals. We have to reduce ammonia emissions by 30%, which also mentioned before. And here's a very rough estimate, which we have done. There's just a technical reduction potential. What can we achieve by using UI in German conditions, and also for the EU, where we have much more use of urea. And so we expect, with this very simple, and also with this EFs as they exist by now, a reduction of the emissions by 6% or 9% for the EU. That's just a rough estimate. And by this, we can also indirectly reduce N2O emissions. A similar picture shows here for N2O emissions. Agriculture is the main source of N2O emissions in Germany, and about 30% of the total greenhouse gas emissions stem in Germany from nitrogen management. That's mainly nitrogen applied to soil. And here again, a very simple calculation of the technical reduction potential by these products. It's based on the actual emission factors, which you can take from meta studies. It's about 8% N2O emission, total greenhouse gas emission reduction from agriculture in Germany, and about 5% for the EU. Talking about urease inhibitors, here's just an older study. There's many meta studies around which show quite similar results. You see that there's a big difference between existing inhibitor products. Mostly studied is NBPT, and we have an efficacy of about 60% emission reduction by NBPT. It's quite different for other compounds here in this study. But there's many active ingredients, in particular also in the German market, like NBPT or 2NP, which are somewhat chemical analog to NBPT, but are less studied, and we don't know too much about it. Similar picture here for nitrification inhibitors. There's also some variation between compounds. Some compounds are studied very intensively, like DCD, much lesser extent nitroperine and DMPP. So some older studies, there's some more data around now, but I think what you can find in recent meta studies is quite similar. And also you see the reduction effects across all inhibitor products in this meta study by Lee, where we also find a reduction of about 40 to 50% of emissions by nitrification inhibitors. But what you also can see here, increase of ammonia emissions if you just apply the nitrification inhibitor without urease inhibitor. And another meta study by Grados about 2 years ago, you can find that use of NIs can be considered one of the most robust measures to reduce N2O emissions based on the actually available data. We have some yield effects. They are less pronounced by both products. It's more pronounced for nitrogen use efficiency, and for yield effects, let's say, you see the error bars very often cross the non-effect line. It's much more vague or more variation there. What I don't show that there's also varying effects for different crops and for different environments. So there's some uncertainty here, and it's these results are mainly from plot trials. Looking at the human health and environmental safety, you can say, at least in the EU, these products are registered under the fertilizer law. It's different in the US, where these are registered under biocide law. And in most industrialized countries, They are registered and toxicologically labeled. Here is a short overview. So several compounds which are already registered and are sold in the European context have the toxicological label of reproduction toxicity. And some of those compounds can be mobile and have toxicity for several organisms. And if we would look at them with, let's say, biocide or pesticide regulatory eyes, some of those compounds would not be registered according to European biocide regulation. Of course, for almost all of those compounds, environmental risk assessments were performed. And but we also find some compounds in surface and groundwater bodies. Also, in particular, some countries also, for example, nitroprene, also in by atmospheric deposition. In particular, it's a triazole, DCD, and nitroprene. And for triazole and DCD, we know it's with high likelihood not from agricultural sources. Triazole, let's say, it's from inhibitors, eventually more from pesticides, because it's also metabolite from pesticides. And from nitroprene, which has been found in US surface water bodies, it's for sure from nitroprene. So that's what we know. What about the unknowns? We say that only a few compounds have been extensively studied. So it's hard, and they're varying in effects. It's hard to derive consistent reduction effects. And it's also unclear whether a continuous application of those compounds can be connected to reduced or disappearing efficacies. There are only a few studies, and in these few studies, we don't this kind of efficacy effect, which you can think which might occur, but only very little data available. What we also don't have is enough knowledge about dose response relationships with these compounds. And so it's hard to infer efficacies when dosages are fertilizer products regulation in the EU. That's very likely to happen with several of those compounds which have to be re-registered. What is also a problem that very often the measurement duration, which for the studies which were included in the meta study, is not long enough to really properly derive an emission factor or efficacy of these products. And so, and if you don't measure long enough, you have probably underestimate the reduction effect. Other way around is that if you consider the unfertilized control, you have, according to IPCC, you have to consider it when calculating the emission factor or the efficacy. In many studies, it's not considered, and so we don't know what effect this might have. And for sure, very little is known and tested for biological nitrification inhibitors in this domain. For human health, of course, I said that a lot has been done, but according to experts, the registration data are intransparent and only partly available. The same applies to the risks assessments. And we know only very limited about the toxicity and the mobility of metabolites and which metabolites exist. And also in different countries, there exist different standards of registration. And so it's hard to monitor the flows. And eventually, some products might come in which are registered in countries which have other registration requirements in your own country. So we don't know too much about the volumes which are around in the environment, so it's hard to assess the effects. And of course, little and much less we know for biological inhibitors. So I want to conclude, eventually do some recommendations. We think that the emission reduction and the agronomic efficacies and the potential contribution to climate mitigation policies is varying between compounds. We cannot just average about across compounds, and it's still uncertain. And so, as I addressed this question already a little bit, we need clear assessment protocols for these kind of compounds, and also long-term application effects is not really properly addressed by now in the data, and soil type and climate effects also the efficacy of these products, which was also presented here in this conference already. For environmental and human health safety, we also think it's not fully assessed, and in particular intransparent, so that we really need. And eventually we need some specific regulation for these products, like similar to biocides. I don't say it must be the same, but we really have a clear understanding what we need to know about these compounds with respect to environmental and human health safety. So we think the use of most compounds, I think some compounds we eventually know enough, is not really ready for environmental mitigation policy. But we think with the compounds, when they are safe and when they're properly tested, can be very advantageous and for environmental and policy and nitrogen policy, but eventually need public support in both development and also eventually for the registration, because the cost burden for these suppliers of these compounds is very high. And of course, we need further research on this, as I've shown. There are several ongoing projects in Germany, for example, our big national project, Nitricrim, and of course, we need much more to know for biological inhibitors. So thank you very much for your attention, and yeah, looking forward to discussion.
You said that we don't know exactly what come up with the facts. And you don't think this. You know, yes, you sure that you guys emissions, but what is the impact on leaching?
Yeah, on leaching, there's some data. I think I showed one slide where it showed the effect on nitrate. Is this slide? Oh, sorry. On nitrate emission reduction. Oh, it's slow, like here. But I'm not sure that the other available data is very limited. It's mainly from grassland and mainly from one compound. And it's in many of these trials here, it's also some kind of pot experiments. And I think it's there the knowledge is really rather compared to the gases emissions, at least in the meta studies. That's
Very quick question.
Yeah.
You mentioned about accommodation and aftermarket effect is smaller. That presumably depends on the mechanism of action. So does that mean you might expect a different accommodation for NDBT versus nitrogen?
Yes, of course. I think the accommodation is probably different for every compound, because they're chemically different. And for NDBT, I don't assume that much of accommodation because it's an extra enzyme. And while for the nitrification inhibitors, you inhibit living organisms. And so it's a different effect. We have also ongoing studies on this, and we find some legacy effects or some variation efficacy effects, depending on the management of these compounds, for the nitrification compounds.
Ramesh, the risk here is let's the responsibility regions as well indicators in action in the way that I supervision can be designed source of.
This is not my slide. Again. It's a surface.
You made it to us, right?
Yeah.
We've got the wrong presentation. Oh, sorry, we have two. Can we have the other talk first? And by the time we can figure out the. Okay. So sorry.
Yeah, that's all. Well, excluded from maximum the nitrogen in possibility.
I have a clock as well. So we'll be verifying the time of this talk.
Yeah, there you go.
Down button.
Yeah.
Okay, ready, steady, go. So I'm going to tell you about nitrogen and cosmology, specifically about nitrogen and ancient cosmology. Why would I do such a strange thing as that? Let's start back a few years ago, and I said to myself in 2005, we're very busy with all this intergovernmental activity, etc., for mobilizing countries. So I asked myself, we need better awareness. Can lessons from history give us some interesting stories? Stories are great, and they help you along in raising awareness. So this is a burning coal cave. It's in Jaharia in India. It's been burning for over 100 years, this coal cave area. Those exist through Central Asia, and the Silk Road went past those. And what happens when the coal burns? It gets sublimation, condensation around the edge, get white crystals collect the white crystals, sell them. It's mainly ammonium chloride, and a really expensive product. Interesting for the story, the price of this stuff was so high, was about 6 to 8 silver coins per 6 to 8 kilograms. I'm getting this wrong. Anyway, I'm going to give you in terms of the price of a human life. One slave could be bought for 6 to 8 kilograms of nitrogen 1,400 years ago. That's how valuable nitrogen was. But even more interesting was that stuff had a name. It's called Nushadra. What is Nushadra? Nush is immortal, Ata is fire. So well-established etymology is immortal fire. The earliest published name for a nitrogen compound is such a crazy thing as that. And that sets you on a trail to look for more, to try and understand why nitrogen would be called immortal fire. You think it might be to do with this Hell's Gate, everlasting burning coal cave. In fact, as I'll show you, it's going the other direction. Up into the stars, above the air is the fire, above the fire is the immortal fire. The stars are made of nitrogen. In their mind, only metaphorically, not necessarily physically. We have a different view nowadays, but nitrogen becomes this whole incredible conversation. In order to get there, you have to go through alchemy. That is really disappointing because alchemy is an exceptionally difficult area. But if you trace your ammonia through alchemy, you find it's there. You also find that alchemy was actually based very close to philosophy. Funnily enough, the Philosopher's Stone was being made by philosophers. What a funny surprise that was. So the Philosopher's Stone is there, philosophers. And so you can further back in time, you find out that, for example, what they did in alchemy, they would marry that material that they call water, and that's actually to do with ammonia, with that material that they call fire, which I don't tell you. And they would marry that material they call earth with that material they call air, and they would marry the products in the great marriage. In medieval manuscripts, you get incredible, exciting images of all this kind of thing. And of course, you've got the gold, how to make the gold. Yes. And how to get immortal life. Yes. Well, remember, there's some things that you can believe and there's some things you can't believe and some things you just have to know how to interpret. But what I discovered was that the fundamental thinking of marriages of alchemy is what you can use to produce the quintessence. You marry them, and the words in Greek, one triumphs over the other, one dominates over the other, one rejoices in the other, delights in the other, were also used in the context of Greek geometry. And now we're on to Greek geometry, finally to the second slide. On the left, you get the publications, the European Nitrogen Assessment. Yes, everybody who contributed to the ENA contains geometry in chapter 5, Platonic geometry. Alkaline air also contains stuff on the alchemy. The peer review paper on this specifically is the geometry of Plato's Cosmos. Hardest ever published paper I had to do, needed 6 editors before they took it seriously. The 7th editor did take it seriously, sent it to peer review, and it got better. Great. And Plato's Puzzle, Hidden Geometry of the Night Sky. Go to the free website, platospuzzle.com, have a party. Now, look at these solids. Those are the Platonic solids. What Plato said, top left is earth, top right is fire, bottom right is air, bottom left is water. He said that if you break them apart, the earth is separate, but if you break the other ones apart with those little scaling triangles, you break them apart with triangles, and one dominates the other and comes together in another. So you can take 2 fires to make an air, forcing them apart, rebuilding them. So that told me that what everybody thought was that the fifth Platonic solid is the dodecahedron. You can't do it with that. But alchemy tells you, yes, you can do it. You're going to remix it. You can make the fifth. So I was left with this conundrum that there's the same language, the same thinking, a geometric analog, an alchemical analog. Therefore, it seemed likely that that dodecahedron was not the end of the story. Because if you take that dodecahedron and you see the little star in the middle, stretch it. So pull that point, stretch. You get another shape. And that way you can do it with equilateral triangles. And here it is, let's stretch it. And you've now done it with equilateral triangles. At this point, you find a really interesting shape because there are 12 main faces of the dodecahedron, 12 months. Yeah. On each of those faces, there are 30 little triangles, one for every day of the month. 30 times 12, that's 360. Plutarch writes about the excitement that this is linked to the calendar. And it's a chance, but it's interesting. They found it interesting. There's my very first one, where I suddenly had a realization when I take this, what if they used that not only for cosmology, which they were clearly doing, but as a basis for a star map. If you put the North Pole here, it divides very nicely into 12, one for every month, one for every star sign. Now, really interesting, if you put the North Pole here, which is the same at that point, and one there, and send the lines down, they split. But because the system doesn't have full 12-way symmetry, odd things happen, and the model becomes very characteristic. So I've separated that model out here, unwrapped it. Here's the North Pole, the South Pole. You go up from those 12 from here, and you come down from those 12 from there, they don't meet in the middle. That forces wide sectors and narrow sectors. And let's just take a couple of bits of evidence. Interesting. This came much later. Servius has this really odd record, says that the Egyptians have 30 days to each star sign, but the Chaldeans have 20 and 40. Nobody has ever explained that. This model demonstrates it very easily. There's our 20, there's our 40, 40, 20, 20, 40, etc. Take another one, which is, we've got 35 seconds, this is impossible. 4 months and 2 months of summer and autumn. You can't explain it, but if you take this, 2 months, 2 months, 4 months, 2 months, 4 months. This is winter, this is 4 months, spring and autumn are 2. All fits on the model. Now I'm going to speed up. Boring scatter graph between the model length of the constellations and the actual length of the constellations. They're not all the same length. Some are long, some are short. The right hand shows you the model measurement comparison. R squared 0.8. This is great stuff. So model performs really well. You get really fun things, funny constellations, not just the zodiac constellation, but others. Eridanus is the river. Some would say it's the River Po in Italy. It follows down that line, turns down this line, turns down that line. They had such a party designing constellations. And where it goes wrong, it even lines up to those significant lines, which is the tropics and 36. So here's the map, new map of the universe. The universe constellations were designed around a zodiac. In their minds, it was a nitrogen compound. The nitrogen compound was immortal fire. So I apologize for taking 23 seconds longer, according to my watch, than I was meant to take. For more information, see the oldest scientific journal in the world, the Philosophical Transactions of the Royal Society of London, which is a great place for a laugh to publish something curious. That gets the alchemy. The astronomy is in these two, and on the website, platospuzzle.com. Open for questions.
That's a wonderful presentation. And you started, and people have also said that telling stories helps people to communicate. And so have you been telling this to people, and has it been helping them to grapple with what is nitrogen, why is it important, why it's a problem?
I like your reaction very much, Peter, because you get it. And stories are interesting. So this is the first presentation of this topic here in India. It's not a first public presentation, and I've yet reserving the fire for a first public presentation on that one. What is interesting is that the reaction is exceptionally negative and not interesting. And the reason, no, let's just play this differently. Depends on your audience. If you put it to astronomers, what you need to remember is that old scientists' main role in life is to prevent innovation and promote their old ideas. So that when you send it to editors, they tell you you're a fool and go away. The follow-up paper to this, which summarizes this, which is published in a peer-review paper, one reviewer liked it. This is British Journal for the History of Philosophy. One reviewer liked it. The other reviewer said, this is so implausible / impossible that it does not need refutation. Okay, so he didn't refute it. He just insulted it. The editor accepted it. We need 2 positive reviews. Go to your next journal. So that's the kind of reaction you get from academia. I think if you know that you're rejected several times and you still believe it, you must backtrack. For the moment, we have to get the International Nitrogen Assessment out. So I need to not spend all my time on this. But when we have it out, I will be pushing more to see if indeed it can help raise awareness on the nitrogen challenge. Can't hear you, Umesh.
You are talking about PV bus and nitrogen. If this is the case, then all plate engines should have atmosphere of nitrogen, which I think all not
This is a very good question. What you have to listen very carefully to is I'm not saying the universe is made of nitrogen. I am saying that the Earth has an atmosphere with 78% nitrogen. I hope you can agree on that, although I doubt that most of you have tested that. And I would encourage you to test the 78% to see if you find it true. What I'm telling you is that the ancients had a philosophical view of the universe that linked multiple dimensions, and the alchemical dimensions of the universe linked to their same philosophy of their geometry. So that in their mind, they saw the stars as being made of immortal fire, which happens to be a nitrogen compound.
Where is the right one?
Thank you very much.
Yep.
Which one? This one?
Yeah, this one.
Reading. This one's final. So sorry. Just a second. Line to wait. एक सेकंड सर just a second, we have to screen share because Zoom पे भी है. It's a hybrid. So sorry.
Hi everyone, I'm Suman Prakash Pradhan from Kathmandu University, Nepal, presenting my work on responses of selected lichen as a biological indicators to nitrogen and metal ion pollution in Himalayan forest of Nepal. Sorry, getting started with the introduction. It is now well-known fact that air pollution and climate change are 2 key worldwide issues, threatening human health, environmental health, and economic growth. Nitrogen is essential component. However, the excessive reactive nitrogen, mainly gaseous ammonia, inorganic nitrogen, and oxides of nitrogen in atmosphere leads to the biological and economic ramifications. Fertilizer inputs and different emission sources giving South Asia the greatest nitrogen input by 2050. Metaline pollution are altering the ecological food web and biodiversity as well. Lichen are well known by indicator. The lichen diversity, lichen functional group, single species indicator, and bioaccumulation are the key scopes of lichens to monitor the nitrogen and metaline pollution in different forest systems. And trace elements in lichens, thalai, are connected to their ambient level. So they are also taken as an indicator of metaline pollution. The global nitrogen use efficiency is roughly 20% and 80% loss to environment, causing severe damage to terrestrial as well as aquatic biodiversity. Most of the studies in past were focused on carbon, which is particularly in the context of Himalayan forest. And earlier studies in Nepal have mostly focused on transboundary atmospheric brown cloud and city level emissions. Only a single research by Panta et al. In 2020 was conducted to investigate the biomonitoring potential of lichens in Nepal. This is for the probably first time extensive work considering lichens, two different lichen species, Usnea and Hypotrochina, that were collected from two distinct forest types of Nepal, were considered in this study to investigate their bioindicative potential. So the main objective of this study is to investigate physiochemical responses considering lichen as a bioindicator for current atmospheric nitrogen and metaline pollution in distinct forest system of Nepal. So based on these research questions, I try to answer my objectives. So what is the status of nitrogen and metaline pollution in Himalayan forest of Nepal? What and how physiochemical changes occur in lichens due to augmented nitrogen and metaline concentration? How do environmental factors influence the deposition of nitrogen and metaline? And then changes in different physiochemical variables in lichens. So these are my two study area. One is rural forest of Annapurna Conservation Area, and one is the urban forest near Kathmandu. And lichen samples were collected from 15 different trees with random stratified sampling strategy. And the Hypotrochina and Usnea species were collected from Purcus, Rhododendron, and Betula trees. And bark and soil samples were collected from respective trees from where lichens were collected. So on-site measurements were done for slope aspect, tree height, etc., which were considered as a environmental factor that influencing the lichens. And laboratory analysis were done for chemical variables such as nitrogen, ammonia, metalines in lichens, physiochemical responses such as chlorophyll degradation, chlorophyll fluorescence, and electrical conductivity. Biochemical responses such as phenolic contents, flavonoid contents, first line oxidative defense as a catalase activities, and second line oxidative defense as a radical scavenging activities were done. For the bark and soil, pH, electrical conductivity, nitrogen, and respective metalines were analyzed. So moving towards the results and discussion, the highest nitrogen and ammonium deposition was observed in Hypotrochina species collected from the Horepani area. However, our study demonstrated the concentration of nitrogen within the threshold provided for Himalayan forest. So, which could be reason for the high abundance of lichen in Horepani area. For the metaline deposition, major cations such as sodium, potassium, magnesium, and calcium were prevalent. And comparatively higher accumulation of metaline was again observed in Hypotrochina species, which was collected from Horepani. And the presence of lead only in Chandragiri, which is in the urban forest, signifies the vehicular emission sources, and presence of nickel only in Horepani, which lies in the rural forest, signifies the household emission sources. So there are many ways to infer these results, such as cadmium and lead occur often together. The presence of metaline in lichen responsible for various physiochemical functioning. For example, excessive level of both copper and manganese can damage cell membrane, degrade chlorophyll, and prevent reproduction in lichens. Here are the results for physiochemical responses. It is suggested that the chlorophyll degradation ratio is around 1.4 for healthy lichens, and higher chlorophyll degradation was again observed for Hypotrochina species collected from Horepani. And the amount of chlorophyll in lichen thalai is frequently correlated with degree of environmental stresses. The moderate chlorophyll A, chlorophyll B, and chlorophyll total content were observed in both site and both lichen species. The highest chlorophyll fluorescence was observed in Usnea species from Chandragiri, and very low chlorophyll fluorescence was observed in Usnea species from Gorepani, signifies the severe damage to photosystem 2. The decreased concentration of chlorophyll A and chlorophyll total were observed in both lichen, along with highest degree of degradation in Gorepani. So the loss of cations in lichen thallus is frequent indicator of nitrogen compound toxicity. Electrical conductivity as a measure of cell membrane integrity represent the measurement of membrane damage and leaching of ions into cells and epitomize the environmental stresses in lichens. Secondary metabolites, mostly phenolic acids and lichen acids, are the key source of defense against different environmental stresses. Catalase is a hydrogen peroxide inhibiting enzyme that produced under stress environmental condition. And the free radicals such as reactive oxygen species, reactive nitrogen species lead to deteriorated health in lichens. So highest and least catalyst activities were found in Usnea species collected from Gorepani and Chandragiri respectively. Here is a principal component analysis for two different lichen species and the physiochemical responses and also environmental variables. So negative impact of nitrogen in physiochemical responses such as chlorophyll, total phenolics, and DPPS radical scavenging was observed, and positive impact of nitrogen was observed in electrical conductivity and catalase activity. And furthermore, moderate to high abundance of lichen and rough bark roughness were observed in both sites. This is the principal component analysis for soil and bark health. Basically, acidic bark and healthy soil were observed, and high bark roughness might buttress the lichen survival in both sites. This is the regression analysis, and result shows the significant impact of nitrogen on electrical conductivity, total chlorophyll, chlorophyll fluorescence, phenolics, and radical scavenging activity. Furthermore, significant impact of zinc on chlorophyll degradation was observed. Similarly, the significant impact of nitrogen on electrical conductivity, chlorophyll fluorescence, and radical scavenging activities was observed in hypotrochinite species. The significant impact of manganese on total chlorophyll was observed, and the significant impact of sodium on catalase activities was observed. There are many factors influencing the deposition and deposition of nitrogen and metal ions in lichens. From our study, we found that ground cover and slope as the most important factor for factors that affecting the nitrogen and elemental deposition in both lichens. Moreover, bark pH is one of the most significant parameters impacting metal ion deposition. On the other hand, wind pattern influences the deposition. This is the wind pattern and source graph. The low wind speed with revolving wind pattern in Gorepani area would be one of the probable reasons for high deposition of nitrogen and highest degree of physiochemical degradation in lichens. And the back trajectory analysis shows the wind flow from Kathmandu was deposited again in Gorepani, which might buttress the nitrogen and metal ion deposition on Gorepani area. The enrichment factor analysis allows us to understand the deposition of metal ion, whether it is from long-range transport or anthropogenic sources or natural sources. Here we can see the enrichment factor greater than 30 in Gorepani area only. So the sodium and calcium were deposited in Gorepani through the long-range transport mechanism, while other metal ions were influenced by anthropogenic sources and local emissions. Here I compared the species efficacy that Usnea versus hypotrochinite species, and I found that the similar pattern, both species showed the similar trend in impact of nitrogen and metal ion in physiochemical responses with R square value of 0.90. So with this premises, I came to these conclusions. The site-specific and specific impact of nitrogen and metal ion deposition was observed in Nepal. Household emission might critically enhance the deposition of nitrogen and metal ion in Ghorepani, and vehicular emission buttress the deposition of these pollutants in Chandragiri. The nitrogen deposition is in critical load provided for the Himalayan forest. The impact of nitrogen and metal ion in physiochemical integrity was observed highest in lichens from Ghorepani area. The physical environment played a crucial role in nitrogen and metal ion deposition. Similarly, the nitrogen metal ion were deposited in lichen mainly from geogenic and anthropogenic emission in both sites. Comparatively, hypotrochina species are hyperaccumulator of nitrogen and metal ion, which might be due to their highest potential of extracellular binding. So I would like to acknowledge UKRI GCRF South Asian Nitrogen Hub for the funding and different organization of Nepal for the research permit and laboratory supports. Here are some of my references, and thank you so much.
Thank you, Suman. I'd like to ask a bit about metals, because I don't know much about metals. And I guess my question is, too much ammonia in the air, and the lichens just won't go there, you get different lichens. So species changes. But with metals, there's a point of accumulation, but are you seeing species change associated with metals, or just a concern about accumulation?
I focused on the physiochemical responses with the deposition of metals, not for the morphological changes in the lichen species. So we did not do any community sampling for which we can convert the result for different composition changes of lichens, but we only focused on the physiochemical responses in lichens.
But do you have a guess? Will you expect to see lichens disappearing with too much metal pollution and different species come in?
Yeah, there are sensitive and both tolerance lichen species. So if they are tolerant to metal ions, they might come up, and if they are sensitive, they might be disappear. Yeah.
Since we're doing in the last slide, we showed basically that there's no difference between species. So they're totally correlated.
Not totally correlated, but somehow they are similar pattern, but comparatively the hyperaccumulator is hypotrochina species, because most of the responses are highest in hypotrochina.
Thank you for the presentation. Just a question about methods. In the region where you was on the legacy, sodium testing and methods.
Sorry, I can't hear you properly.
I assume this is an atmospheric position, right?
Mhm.
Sodium and some methods.
Yeah.
What was the source of origin of this?
Source of, for that, we did the industry factor analysis for that. So for calcium and sodium, there are source from the long range transport mechanism, but we did not do any source ascertaining in the field. So we can't just answer this question. Yeah.
Thank you.
And thanks for the nice talk about metals actually. I was curious because we were talking about atmospheric deposition. Did you by chance also mention mercury? Because when we talk about atmospheric deposition, I maybe think of one metal which is quite relevant. Maybe you can just see it.
No, we love to see it, but unfortunately we did not do mercury analysis. Yeah.
Okay, thanks everybody for one.
I'd like to now request the co-chairs to please felicitate the speakers. Okay, Dr. Peter Brown.
Thank you very much.
Dr. Alexandra.
Giving this out. You're completing that. This.
Dr. Hannah. Dr. Andreas Kapos. Dr. Sundar. And Dr. Mark Sutton. Dr. Mark Sutton, could you please? And now I'll request Dr. Andreas Popolski to please felicitate the co-chairs.
Thank you very much. Everybody. All the speakers in one row, start to the left and then.
I request all the speakers to please stay for a photograph.
Around.
मेरी सनस्क्रीन क्यों नहीं हो रहा? अबे तू मत
दिक्कत है भाई। Find people also.
अभी बहुत देर देखा है।
Sorry.
सनस्क्रीन क्यों नहीं हो रही थी?